Sorry. So, good morning, all. Welcome back to the last day of our conference. We had three eminent speakers this morning. Our Professor Parthasarathi, Professor Ashwini Parik, and we have one online concluding session by Professor Fire also. So, chairing this session, we have. I'm not standing in between because if I speak more, that will ultimately limit the time of the speakers. So we have Dr. Deepu Matthew for chairing this session. He was professor at Center for Plant Biotechnology and Molecular Biology, our agricultural university, Thrissur, and he has been a prolific researcher, and he is a member of fellow of the NAS, National Academy of Agricultural Sciences, New Delhi. He is fellow of Indian Society for Plant Physiology, and as well as member of National Academy of Science, India. So I welcome Dr. Deepu to the dais to chair the session. Okay, thank you. And he also have developed some varieties of crop plants and of KU paprika, one variety, as well as another resistant variety of okra linus also has been developed by Dr. Deepu. And for co-chairing the session, we have Dr. Sumesh. Earlier he worked at Rubber Research Institute, Kottayam. Now he is working in the Agricultural University of the Neeleshwaram campus. So, welcome Dr. Sumesh. And for as rapporteur, we have Dr. Fasila. She was in fact our former student of this department. Now she is working as assistant professor at neighboring college, in Unity Women's College. Welcome, Dr. Fasila. So, I think the chair will welcome the speakers. Thank you, and all.
Thank you, sir. Yeah. Respected professors, scientists, research scholars, and dear students. Today, we are entering the third day of a very beautifully organized international conference. And usually we know in every seminars, there will be one or the other shortcoming. Even after doing everything for a very smooth conduct of the program. But this one is really exceptional. And I have no words to congratulate Dr. Puttur and his entire botany team for organizing such a very beautiful seminar. Yeah. Now, we are going to start with the final session. And in this session, we have three eminent speakers, very eminent speakers, who are going to talk to us on very important topics. And the first speaker is Dr. Parthasarathi, a huge figure in the field of plant physiology in India. And he will be talking to us on compatible solutes, fine-tuned carboxylase or oxygenase activity of Rubisco as a strategy for adaptation to abiotic stress. From the brief CV given here, I can understand that this is a very favorite topic for him, and he has worked a lot in this area. And to introduce, as I know, there is no meaning in giving an introduction because he is already very well known to all of us. Anyway, to have a gist on his profile, Sir is a professor in the Department of Environmental Sciences in the Andhra University. And in the month of July, last July month, he got retired from the University of Delhi after a profound service for 22 and a half years. And also, previously he has he was teaching in the prestigious universities and colleges like Jamia Millia. And he received his MSc, MPhil, PhD, and everything from University of Delhi. And his research interest goes like elucidating the basic facets of adaptation of plants to the abiotic stress, plant microbial interactions, isolation of fully functional genes, as well as the synthetic genes, and it extends up to the nanotechnology. And he has 140 publications, 3 international patents, and 2 Indian patents, and also his team has immensely contributed to the mechanisms involved in fine-tuning the NADP as well as NADPH ratio, and also the roles of the compatible solutes like a proline, glycine betaine, the sugars in regulating the carboxylase and oxidase activity, and so on. And he also has received many awards. To name a few, he received the National Bioscience Award for Career Development 2000 and 2001, and fellowships from Japan, grants from the Research Institute of Innovative Technologies for Earth in Kyoto, and also the Indo-Japanese, Indo-Swiss, and also the AP Netherlands fellowships. And with these few words, I invite the very eminent speaker and invite you all to hear from the legend. Thank you, sir.
Thank you so much for such wonderful introduction. Anyhow, I'm just a small teacher like you. It's not that all of us are almost at similar level. Maybe only thing is we are at different hierarchical levels. Now, to begin with, I just wanted to share a few of the photographs where basically Professor Govindji has been visiting our team at regular intervals. And you can see the manner in which he sits with scholars and explains. So that is the simplicity with Professor Govindji Burgess. And the lower one is one of the workshops which we organized at Delhi University, and where Dr. Vanaja has also participated. This is with some of the participants who are there in that. Okay. Today I'm going to put forth before you certain things which I learned along with my research scholars. The focus of today's my talk would be basically on compatible solutes, the relation to photosynthesis. I'll be focusing more on Rubisco activity, and I'm basically more concerned about carboxylase to oxygenase ratio, the maintenance of this. So before I go further, it is my duty to let you know that these are the people who are behind educating me in the field of compatible solutes. We have Prasanna Mohanty, who has contributed significantly in my knowledge in the area of light reactions. I think ours is one of the teams which has wonderful collaboration with him, and we have about 15 papers published with his team. And of course, he has been a great mentor. He's very particular about the experiments and the repetition of experiment, and also repeatability of the results. The second great person I always appreciate, whom I consider to be one of the best plant biochemists India has ever produced, that is Professor R.C. Sachar. He basically superannuated from Delhi University, and then, of course, he is the person who has done very good work in the area of Rubisco. Very simple and down-to-earth person. And of course, we are having Govindji and Professor Rito Strasser. Both of them have been visiting our laboratory, our team. And we have learned quite a bit from both Professor Govindji as well as Professor Rito Strasser. And in the center, I'm having the man Professor Mohammed Amin. He is the person who is behind my success. He is the person who has encouraged me in developing my research activities with freedom. He has given me complete freedom. And below, we are having the scholars, like Alia is my first PhD student. Alia and Sandeep, Sandeep is now, Alia is in Germany, she's a professor there and now working in neuroscience. And Sandeep Arora is basically a professor at GB Pant University. He is now dean. Dr. Shamila, she's my wife also, and she has been associated with our team for almost right from the beginning till date. And of course, we are having K.V. Prasad. He's basically, he's present in US. Shiva Kumar is the person who has contributed significantly on Rubisco along with Shamila. And then we have Vani and Jose. Alia, Sandeep, Vani and Jose have done very good work in the area of photochemical reactions. And of course, they have also done some work related with Rubisco. We have always been talking about this. This has become a very favorite slide. We always try to talk about the productivity. We always try to talk about how to take care of this particular thing. We are talking about the attainable one, the actual yield. Now, how can we basically manage? If we manage this, if we really need to manage these particular things, we need to ensure that we are providing ideal conditions for ideal photosynthetic activities. Now, when we are looking at this particular thing yesterday, we have heard quite a bit regarding below ground activities. And of course, the good work done by Professor Sabala and Professor Babu have been just wonderful. It's something remarkable work they are doing. They're trying to identify some of the factors in the root system which could be contributing towards the yield factors also. Now, that is something which we should try to look forward. That means below ground biodiversity, besides the root system, is going to play extremely important role in handling this problem of yield attainability, whether you are interested in having, if you want to really take care of the yield, it's important for us to look into many of those important features. Now, coming down to the topic of today's lecture. So obviously, when we are looking into, I'm basically going to refer to this. I consider this particular thing as the most important industry nature has invented for every one of us. Now, without this industry, you cannot imagine life. You cannot imagine synthesis of carbon skeletons. I projected this as simple industry wherein it is basically taking in carbon dioxide and using water, and the basic input we are basically providing, the energy input, the fuel which it is using is the solar energy which is abundant in nature. And what is the output we are getting? We are getting beautiful outputs. We are having carbon skeletons, although we may talk about sugars, but you think this is the thing which is going to lead to the production of all sorts of carbon skeletons. Let it be glucose, or let it be, talk of any carbon skeleton. Whether directly or indirectly, this is going to be coming from the carbon resources which are going to be coming from here. Of course, we are getting oxygen, which is again essential for survival of all of us. The beautiful thing it is doing is, it is basically taking out the pollutant, carbon dioxide, which we are considering to be pollutant. It is basically removing the pollutant and converting this so-called the pollutant into a useful product. Now, obviously, when we are looking into this industry which I talked about, it's basically having two wings. The wing one of this industry is responsible for harvesting solar energy. This is the thing which is basically capturing the energy associated with the photons. And then it's energizing the electrons. It's basically the thing which is responsible for production of assimilative power. It is basically converting, it's basically using water as a resource for, it is drawing electrons from this particular thing, and obviously it is energizing the electrons and finally providing these electrons to NADP so that we can get reducing power, that is NADPH, and simultaneously it is producing ATP because it generates proton gradient. And in addition, of course, we are getting oxygen out of this. And these are all biophysical processes. Now, this is the jet scheme which we always talk about. All of us are fully aware of this jet scheme. If you look into this, obviously we are talking about the photosystems and cytochrome F complexes. Now, these are the things which are together responsible for, of course, along with the quinone and other components which are there, plastoquinone and other components which are associated with them. These are the things which are basically converting, energizing the electrons. And we are already aware of this particular thing. Let me not spend too much time on this aspect, but the only thing which you should try to learn is, especially for students, imagine the extent to which electrons are basically being energized. These photosystems, this machinery is basically increasing the energy associated with electrons by somewhere around 26.2 kilocalories per mole of electrons. The second wing, which I'm going to focus today, is related with the second wing is the thing which is basically using the assimilatory power which is produced by wing 1 of this industry, and then using carbon dioxide, fixing carbon dioxide. And obviously, this machinery or this wing of photosynthesis is nothing but it is having various enzymes. The machines are made up of various kinds of proteins, various enzymes which are present there, which are basically helping in converting carbon resources from one state to other state in various ways. And of course, we are basically referring to Kelvin-Benson cycle. So when we are looking into the thing where we have our focus of this conference is on stress. So we know that stress has negative impact on photosynthesis. Now, I would like to stress how exactly the plants are handling this particular thing, how exactly plants are trying to counter the stress. Now, one of the things which we have been always trying to focus is on compatible solutes, and of course, fortunately, I had wonderful students who have contributed significantly in making me understand details about detailed mechanisms that are involved in the way compatible solutes are produced, the way compatible solutes are basically associated with imparting stress tolerance. Now, one of the molecules which we worked was on proline, and of course, I think I have maximum publications in the area of proline. And of course, this is something, when I'm referring to compatible solutes, I'm referring to the organic molecules, it's small organic molecules, which are basically accumulated in living beings. Now, obviously, as an adaptive strategy to counter abiotic stress. The most important thing, we are using the term compatible. When I'm using the term compatible, I'm basically stressing on the point that they're not going to perturb any of the essential metabolic reactions, even when they are present in very high concentrations. So when we look at something like the proline, initially it has been discovered. When it was discovered, it was believed that it is associated with osmoregulation. There are people who believed that they protect proteins and enzymes. It's known to help in regulation of cytosolic pH. They're going to help in scavenging reactive oxygen species. They protect photosystems, and they regulate NADP to NADPH ratio. So, of course, as per first report, it is basically contributing towards the osmotic stress tolerance. And these are the three scholars who have contributed significantly towards the initial part of our success on compatible solutes, the Alia, Sandeep, and Prasad. Now, these people have basically shown that even mineral deficiency stress can impose, it can make plants to produce proline in sufficient quantities. So that means you are able to find proline production even under situations where there is no osmotic stress at all. And second one is, even in presence of heavy metal stress, you are able to see proline accumulation. When we talk about heavy metal stress, what is the concentration of heavy metals we talk about? We talk in millimoles, we talk about micromoles. So these millimoles and micromoles are not going to impose any kind of osmotic stress. And we have demonstrated that the proline can get accumulated even under UV radiation stress. Even when you expose them to gaseous pollutants, you see the proline accumulation. None of these particular things, whatever things which are reported in these 4 cases of stress, none of these things are anything to do with osmoregulation. In spite of that, you are able to see this. So therefore, my basic question, and of course we have always been questioning this, is stress-induced accumulation of compatible solutes really related with osmotic adjustment? For everything we always try to say, Proline is getting accumulated because it is going to take care of osmotic potential, osmotic stress. So is it really involved in that? So let's try to look into the thing. We wanted to disprove this particular thing that it may be associated with osmoregulation. We don't deny that particular part, because anything which is soluble in water, that is going to impose osmotic regulation. Anything which is soluble, let it be ions, let it be sodium ions, let it be potassium ions, anything which is soluble in water, that is going to contribute towards the osmotic strength. So therefore, we wanted to perform very simple experiment. We somehow believed that it could be disturbing the electron transport system, because we have already started working with Professor Prasanna Mohanty, and we have also been working with mitochondrial electron transport. Therefore, we wanted to check whether electron transport is anything to do with disturbance in electron transport, is something to do with accumulation of proline. So we have performed very simple experiment. We have exposed plants to various kinds of inhibitors, which included mitochondrial electron transport inhibitors. We have used rotenone, we have used antimycin A, we have used casein, potassium cyanide, casein. And along with this, we compared that particular thing with the proline accumulation under sodium chloride stress and cadmium chloride stress. And we had the control. You can just see the extent of enhancement. This is within few hours. And then, when you try to compare with the proline accumulation with NADH, there is a direct relation. So under stress, what exactly we are expecting is, what exactly we are observing is, we are basically finding increase in the level of NADH. So we are not saying that the quantity, it is going to get accumulated. Please remember, for the sake of students, I'm stressing on this particular thing. Never say that NADH is getting accumulated. It cannot get accumulated. Only thing is, the ratio of NADH to NAD is getting altered to a significant extent. The bulk of NAD plus NADH, it will be maintained to some extent. Fine? So only thing which I'm stressing is, the NADH component of this pool is going to be much higher when compared to NAD pool. So therefore, nature is beautiful. Nature has invented so many things. Nature has invented so many different mechanisms to handle this problem. Now, one of the things which it has done is the evolution of dehydrogenases. It has evolved dehydrogenases. You look at the beautiful examples which you have been learning right from schooling. If you go back to our schooling, we have been teaching scholars that fermentation is important. We talk about, when we are talking about fermentation, obviously it has some important role to play. So you heard of alcohol dehydrogenase. You heard of lactate dehydrogenase. These are very common things which you basically hear, especially at school level itself. We hear about alcohol dehydrogenase as well as lactate dehydrogenase. And in addition to these dehydrogenases, we are having malate dehydrogenases. We are having malate dehydrogenases, and of course, glycolate oxidase. This is also able to oxidize. All these dehydrogenases which we are having, they are able to oxidize NADPH or NADH to NADP or NADP. NAD or NADP, fine? So that means it is converting reduced state of these particular things into oxidized state. And that's what is important. We'll try to learn why is that important. So we believe that proline accumulation is again something to do with regulation of the ratio of NADH or NADPH to NADP or NADP. NADP or NAD, right? I tried to show the simple reaction of this. Proline is basically synthesized from something like glutamic acid. And glutamic acid is converted into proline in 2 steps. We're having in both the steps, you are seeing the conversion, you're seeing the oxidation of NADH or NADPH to the respective states. You are basically finding oxidation of NADPH into NADP, NADH into NAD. Same thing is happening even in this. So that means for each mole of proline that is being synthesized from 1 mole of glutamic acid, what exactly we are able to find is, you are able to find utilization, or let's say conversion of 2 moles of reducing power from reduced state to oxidized state. So that is very important for us. Now, at the same time, another beauty as far as this reaction is concerned, when you look at glutamate, glutamate is having 2 carboxylic groups, whereas proline is having a single carboxylic group. So one way, besides handling this, it is also trying to help in buffering the cellular pH. It's also trying to help in buffering system. So is proline a good buffering system? Is proline, the proline synthesis related with maintaining cellular pH? One way, obviously, by using reducing power. Second way, you are also going to see this particular thing. You are having 2 carboxylic groups over there and compared to a single carboxylic group which is present over here. So when we realize that proline is basically being synthesized in the chloroplast, the basic question which we had in our mind is there are reports that proline may be getting accumulated in chloroplasts at concentrations as high as 1 molar. 1 molar is very high. So therefore, the basic question which we had in our mind was to see whether the photosynthetic events that are taking place in chloroplasts, they will be getting influenced by this or not. Because that's the most important metabolic event taking place over there. So therefore, we entered into that business of this. That is the time I basically entered Professor Amonti's lab. And that's the first time we started working in collaboration. And of course, he was very kind. And then that was the time when we started this work initially. And then obviously at that time, Alia joined as a PhD scholar. And she has taken over that particular work which we have initiated with him. And then we could show, we could demonstrate that proline does promote both photosystem 2 as well as photosystem 1 activities and even whole chain reactions. We tried to test this particular thing not only in one plant system, we tried to test this particular thing in multiple plant systems. And we are able to demonstrate that this particular thing basically protects, proline also protects photosynthetic machinery against photoinhibition. You can just see in presence of proline, if you try to compare in a control thing, this is the control one, right? The one which is, this is the one which is in presence of proline, in absence of proline. The above two are basically under dark conditions, therefore let's not bother about that. You should just basically compare these two things. You can just see the difference. When you're imposing stress, it is getting rapidly decreased, but in presence of proline, you're able to find significant curbing in this decline. Okay, that means it is trying to, proline is definitely helping in protecting photosystems against photoinhibition, photosystem 2, we focused on photosystem 2. And then obviously, we try to find out what could be the reason behind that. We have clearly shown, demonstrated that it is basically, it is in a position to either scavenge reactive oxygen species. There are two possibilities. The second thing is, it may also be helping in reducing the production of this. If malondialdehyde level is going down, it doesn't mean that it is simply scavenging. There's every possibility that production of this particular thing may be getting curbed to a significant extent in presence of proline. So the next thing obviously was for us to see what is happening to the dark reactions. So obviously my interest was to look at Rubisco. I was very fortunate to have alumni of this university, Shiva, wonderful biochemist. Again, it's very difficult to get a scholar like that. Very hardworking. I have seen him working in the laboratory for almost 48 hours continuously, of course, with small breaks in between. So he used to purify the proteins, and he used to make us work like that also. And he always had, of course, many of my scholars always put me on toes, made me to learn more so that I can resolve their problems. So that is the beauty as long as Shiva is concerned. And of course, Shamila has been working along with us. This is the time where I approached Professor R.C. Sachar. He is retired from there. Huh? Oh, okay. We, we, uh, so we approached Professor Sachar, and Professor Sachar has basically, he has kindly agreed to help us, because initially we had some problems when we tried to measure Rubisco activity in presence of proline. As far as that time when we were looking at Rubisco activity as a carboxylase. Because I'm more concerned about carboxylase. So I was taking this as RUBPKs. So therefore, when we initially did this, we were able to find decrease in the activity of Rubisco, carboxylase activity of Rubisco. I didn't believe that. I thought we were doing something wrong. So therefore, we approached, that time, of course, Shiva didn't join. That was before Shiva joined, we did this. But when I approached Professor Sachar, he said, okay, Partha, there could be something wrong. Then I requested him to visit our laboratory. He came in person and he trained us. He trained Sharmila and Shiva. Both of them got trained extremely well. Of course, we used to go and pick him up. And he was very kind. He was a wonderful teacher. And another very important feature which I find as long as this great man is concerned, he is the only teacher I have come across across the world, I can say, who basically wanted the welfare of the student, 100% welfare of the student. When we were getting good results, when we wanted to put his name in papers, he said, Partha, please don't do that. You came with your idea, you have done certain things, please don't put my name even in acknowledgement. People will not let you come up. They say that you got these particular results because of Professor Sachar, it's not your contribution. You don't do that. He didn't let me put his name even in acknowledgement. That is the highness of a great teacher. Okay. When I'm talking about Rubisco, obviously, we're always concerned about biomass. We're basically concerned about the amount of carbon that is being fixed. So what kind of things, how exactly it is distributed? Now here, obviously, we are talking biomass in terms of gigatons of carbon associated with each group. Now, as is evident, majority is obviously associated with plants. If you're interested, these are the two great men. I've come across few papers of them recently. These are from Weizmann Institute of Science, Israel. And of course, you should see these papers. There's a lot to learn from these papers. So, as it's evident, majority is obviously associated with plants. Now, I'll just try to compare some of the things based on this paper. Now, obviously, when I'm looking at Rubisco, we are basically talking about something which is basically helping in carbon assimilation. It's basically helping in storing carbon in the world, producing carbon for maintaining living beings. Now, we are basically referring to, obviously, the enzyme, which is having a bifunctional role. It's acting as both carboxylase as well as oxygenase. And obviously, this is the only, although they said that it is responsible for the vast majority of global thing. Maybe I'm not aware of any other thing which is fixing global carbon fixation, which is responsible for global carbon fixation. There could be something which I'm not aware of. And of course, it is the most abundant protein on the earth. Please remember, it's most abundant protein on the earth. If you try to compare the biomass of Rubisco with the biomass of the human beings on the earth, you find this to be much higher. The biomass, total biomass of Rubisco on earth is 0.7 gigatons. Whereas, total biomass of entire human population, so that is today standing somewhere around, it's almost nearing 8 billion. I was looking in the morning, it was about 7.98 something. It's almost reaching 8 billion. If you look at the thing, biomass of the total human population is less than the total biomass of Rubisco, which is present on the earth. So 90% of this rubisco is found in the leaves of terrestrial plants. And in general, when you crush a leaf and you look for the protein, 3% of total protein in the leaf and 65% of soluble protein in the leaf is going to be represented by nothing but rubisco. I'm talking about 65% of total soluble protein and 3% of the total protein. So they are basically going to be represented by rubisco. And of course, this has been first purified by Wildman and Bonner way back in 1947 when they discovered this particular thing, when they are purified, they named this particular thing as fraction 1 protein. So for the sake of proteins, students have put this term fraction 1 proteins. You people should not have any confusion, especially in some of the competitive exams. People may ask this question, so you should be aware of this. What did I play? Sorry. Okay. So when I look into this rubisco activity, obviously, the carboxylase activity, as I specified, it is going down. Whereas oxygenase activity of the same rubisco is increased under stress. When you impose stress to plants, the carboxylase activity drops down quickly, whereas oxygenase activity increases. So this is the thing which was very baffling for us. When we are finding a decrease in carboxylase activity, of course, to begin with, we are looking at only carboxylase activity. I'll come back to this story a little later. And then let's see, in case when carboxylase activity is getting dropped, what is happening? The carboxylase activity, if rubisco activity is going down, obviously, there would be significant decline in utilization of NADPH. So that means there will be buildup of NADPH to NADP ratio. The ratio is going to increase. The reduced state will be present in very high concentrations. So this is going to disrupt entire metabolism. In a normal cell, in the morning when photosynthetic events begin, in the early morning, you might be finding majority of NADP pool in an oxidized state, as you can see over here. So when you progress, what happens? Obviously, under stress, the majority of NADP pole is in reduced state. They are all present in NADPH state. That means what exactly we are emphasizing is there is a significant increase in the ratio of NADPH to NADP. So that is the thing which is leading to the generation of superoxide anion radicals that basically generates, leads to the production of superoxide anion radicals. And you are aware of what are the consequences of this particular thing. Again, let's not talk much about this particular thing. And of course, all of you are aware of how exactly ROS are being generated. And this is the thing which is basically going to lead to the next step is obviously the reason I included this slide is this is something which has been prepared along with both Professor Strasser and Govindji. And this obviously we do expect photo inhibition. And as a result of stress, you do find this kind of thing. It's more because of the disturbance in reducing power. So coming back to the story of this, as I just said to begin with, we have found a decrease in the carboxylase activity when we are exposing RUBP to proline. Look at the way the proline is influencing carboxylase activity. No doubt, carboxylase activity, I showed earlier slide where I said the carboxylase activity is affected significantly by stress. But here I am talking about impact of proline on carboxylase activity. Proline, which is supposed to be otherwise protecting enzyme activity, protecting the proteins and the entire machinery in the cell, as far as carboxylase activity of rubisco is concerned, we are finding a sharp decline. We thought it may be associated only with one plant system. We initially worked with Indian mustard. And then we moved to Sesbania. And that is the time Professor Sajjar was playing very important role with us. And both Sharmila and Shiva, they have just been working on this particular thing. They used to get the materials and they used to do this. And of course, we had Brassica Gensia, Sesban, and Borregia satae, we tried in rice. In all the three systems, we got almost similar results. So that clearly showed that proline is having some negative impact on carboxylase activity, although it may be less when compared to the normal salt stress. So, but we are finding a negative impact. So this is a very baffling thing. As a physiologist, I could not accept this. We are saying from one side, We have demonstrated, we have published papers saying that it is protecting photosynthetic machinery. PS2 activity is basically being protected. It is curbing the ROS. ROS production is able to scavenge. But here I am showing that carboxylase activity is going down. So it is something which has, we could not digest, but we published it. And Professor Satya said, Partha, there's something in this. You don't wait, publish. We waited almost for 1 year, and then we simply published. The moment we published, within a week's time, we got acceptance letters. That's from BBRC. And then we wanted to see. We didn't just stop that. Of course, negative results also many times are very useful for us. They're extremely useful for us. So what we have done is, we have increased the concentration of this particular thing up to 3.5 molar we have tested. Even when we have exposed Rubisco to concentrations as high as 3.5 molar, we are not finding complete decline in carboxylase activity. The carboxylase activity, at least 30 to 40% of carboxylase activity is still remaining. Generally, when you're under osmotic stress, any kind of things, what will happen? You should have gone down completely. But here we are at least having 30 to 40%. So this gave us an idea that, and of course it is known in literature, that Rubisco obviously is an oligopolymer, oligomer. It is hexadecamer. It's basically made up of 16 polypeptides, 8 smaller subunits, and 8 larger subunits, as I showed in that particular picture. It's basically a huge structure. So we thought, and it is also known, that the catalytic sites are associated with LSU, larger subunit. And the function of smaller subunit is to help in promoting the activity. Okay? So therefore, we then thought that if there is a possibility that proline might be dissociating smaller subunits from larger subunits. We know that it is known in literature by then that smaller subunits enhance activity at least 2 to 3-fold. Then we thought this could be because it may just be dissociating the smaller subunits from larger subunits. So then we performed some very simple experiments. Now obviously, we have gone for, the next thing is using electrophoretic techniques. We just went for that. And it has given us a very clear-cut clue. So the upper one basically is native gel electrophoresis, lower one is SDS. So as you can see, in control and NaCl treated one, we're having a single band. Whereas in proline treated one, we're having 2 bands. The first band, we presume that to be L8, and second one is S2 units. So that means 2 smaller subunits, and there you're having 8 larger subunits associated with them. So then what we have done is we basically pulled out this protein, we eluted the protein, we basically have eluted this, and then we have done SDS. When you have eluted this control and run SDS, we are getting 2 bands, larger and smaller subunits. Same is the thing with the one which is treated with NaCl. Now this is the one where we have taken the upper band. When you take upper band and when you're going for SDS treatment, then you're having a single band here. The lower one has given a single band over here. And again, the BBRC accepted our paper instantaneously, even on this particular thing when we talked about the mechanism. What exactly is happening in this particular thing is obviously you're having a oligomer, you're having hexadecamers sitting on the top. When you're treating the proline with this particular thing, it is basically helping in dissociating smaller subunits from larger subunits. And then we're basically getting up in this. And you cannot recover them. You cannot make these particular things to reunite even if you remove the stress. Because it's not easy. Under artificial conditions, it is very, very difficult for us to reunite these because we still not understood completely how exactly these are getting assembled. You require some special chaperones and chaperonins, and I don't think anyone has so far able to discard that particular thing perfectly. Okay, as I specified, that we're having one side an increase in oxygenase activity towards other side decrease in carboxylase activity. So we have basically now tried to focus on oxygenase activity, carboxylase activity, we have already said it is going down. So our job is next to look at what could be happening to oxygenase activity. This is the thing which is a wonderful observation we made. I think this is the first observation which is made by anyone in the world. We have reported that, of course, we have seen, this is a very clear-cut thing. In presence of proline, you are able to find 100% decline on oxygenase activity. Whatever oxygenase activity is there, the increase in oxygenase activity is completely curbed. It's completely alleviated in presence of proline. So this is the first time we have reported that proline completely curbs, it completely alleviates oxygenase activity of carboxylase. In the process, what is it doing? It is basically helping in maintaining high carboxylase to oxygenase ratio. That's what is maintained. That's what is most important for production of good biomass. You need to be ensured that you are able to produce, you're able to have good carboxylase to oxygenase ratio. That is most important. And that's what we are able to demonstrate in this particular thing. And instantly, of course, this is the thing which has satisfied me very well. At least I had some answer to give to the scholars. Why is proline being thing? And of course, now we are 100% sure that it does help in maintaining good carboxylase to oxygenate activity. This is one of the adaptive strategies too. So then, of course, we didn't leave with this. That was a proline story. Then we wanted to test with other compatible solutes. This is the case with glycine betaine. We have done wonderful work. We have produced a lot of transgenics related with this. We have done a lot of work on this. Again, I'm not focusing on all that. I'm just trying to focus only on oxygenase activity, carboxylase and oxygenase activity of Rubisco. So what we have basically obtained is, as far as glycine betaine is concerned, this is a beautiful compatible solute. It is not declining carboxylase at all. It's not having any negative impact on carboxylase activity. Rather, it is promoting carboxylase activity. Of course, I won't say to any great extent, but it does promote. It's not. But as far as oxygenase activity is concerned, you look at the way it is completely collapsing, just like proline, even glycine betaine also has potential to curb that. Same thing you are able to find over there. And then we have started looking at other compatible solutes. Sucrose is a beautiful compatible solute. Sucrose, which is present in the plants, and I'm sure students should know this. Why is sucrose being preferred by plants? One of the reasons why it is preferred by plants, and even if it is present in high concentration, it is not definitely going to have any negative impact. And that also completely cuts down the oxygenase activity, and it does not do much anything. It's not having any kind of negative impact as long as carboxylase activity is concerned. None of the sugars have, except for sorbitol and to some extent glycerol. But sorbitol has some negative impact. Rest of the sugars which we have tried, they do have. They don't have any negative impact as long as carboxylase activity, but however, all of them basically curb oxygenase activity, both under normal condition as well as under stress conditions. Even in presence of stress, you will be able to find complete decline in the oxygenase activity. Just look at the way they basically curb oxygenase activity. So then obviously we were trying to now look into things. I won't stress much on that. It is still in the preliminary stage. We are trying to understand, and of course, we would like to invite collaboration from anyone who is interested. We have done some preliminary work. We believe that, of course, we are trying to understand the way these compatible solutes could be influencing Rubisco activity. So we initially looked at the place where carbon dioxide, of course, we try to dock with these molecules, and then we know where are the things these things are binding. And we believe that just like, for example, this is carboxy arabinitol phosphate. This is a very good inhibitor of Rubisco, that is, everyone is aware of that particular thing. As far as proline is concerned, proline is supposed to be basically is not helping in attachment of oxygen. What proline is doing is it is dissociating. But as long as we are basically concerned about larger subunit. When I'm looking into the larger subunit, it is not letting, it is binding to the portion where oxygen binds. So therefore, oxygen will not be in a position to bind to this. So same is the case with these compatible solutes. When you look into glycerol, or mannitol, inositol, sorbitol, sucrose, trehalose, or even glycine butane. It is binding to the sites where oxygen is binding. So that means there is a possibility that all compatible solutes, they might be having certain portions which are going and binding with Rubisco and influencing Rubisco activity by not letting oxygen bind to them. So I would now like to summarize. If you basically look into the thing, whatever we test. So nature has invented beautiful mechanisms, wonderful mechanisms nature has invented. Only thing is, we are trying to understand. And it's impossible for any of us to really come closer to what nature has done for us. Now, let's try to understand those particular things, try to see, we can try to unreveal some of those particular things, try to use aptly for the human welfare. Now, as far as today's thing is concerned, my basic concept, basic emphasis has been on how to manage NADPH to NADP ratio. If you're able to handle NADPH to NADP ratio or NADH to NAD ratio, you can do wonders. You can take care of the biomass production, you can take care of all stresses without any issues. But only thing is, we have to know how to manage that. Can we build a plant system where the rate of reduction of NADP by solar energy mediated photosynthetic electron transport exactly matches, or it perfectly matches with the rate of its oxidation by Calvin-Benson-Bentham cycle? Is it possible for us? The day you do this particular thing, the day you will be able to achieve this, you will be able to take care of the world hunger. We will be having excess. We will be having much more than what is required for the production of energy. Whatever you do, in terms of food, in terms of energy requirement, or whatever fuel requirements you talk about, everything can be taken care the moment you are going to take care of this. Now, is it possible for us to increase carboxylase activity of Rubisco? And this is the one something we are now talking too much about genome editing. I somehow feel that is something which the genome editing people should try to focus on. Okay, we should try to identify the goals. We should try to know what is the thing which we require. So according to that, we should think of where exactly what nucleotides, which portions have to be handled so that we will be able to edit that particular thing appropriately. That's the most important thing. I think that is going to resolve our food and energy crisis to a very significant extent. Now, is it possible to divert reducing power for some other events, some other anabolic events, something like production of some carbon skeletons? Can we plan to produce some hydrocarbons which will be very useful, that can be used directly as a biofuel? So these are the questions I have. And with this, I would like to end. Thank you so much for your patient hearing.
Thank you so much, sir. It was indeed a great talk as expected. And the topic is open for the discussion. Any doubts? Yeah.
Yes.
Great talk. It refreshed a lot of this photosynthetic memories from the student life. Yeah. So I have two questions regarding this proline system.
Okay.
So you showed that under stress conditions, proline synthesis is very high, up to even more than 1 molar. That's a lot. So, and proline.
They are the reports.
Yeah, and proline synthesis is happening from the glutamate, glutamic acid, glutamate.
That is the major one.
So glutamate is the first amino acid synthesized from the nitrogen assimilation.
Perfect.
So that means under stress condition, to synthesize that much level of proline, there is a lot of disturbance in the carbon-nitrogen partitioning in the plant. And in that way, have you checked under nitrogen limiting condition, what happened? Under stress, plus also limiting nitrogen conditions. So that's the one question.
Let me answer this. Okay. Now, as far as you're talking about accumulation of 1 molar proline, that is a report.
Yeah.
Okay. But whatever we're talking, we always talk in terms of nanomoles per specific weight. Okay. As far as your nitrogen limiting conditions, we have never tested that. But the other component which you're talking about, something like diversion of something from one place to other place, it may not really play that important role. It's not that we're not talking about that high concentrations. We're talking about how much. It's not going to be that high. So it's trying to, first, it's trying to survive. It's trying to get adapted to something which it might kill, something which might, so it is basically adaptive strategy. It's trying to do that. It's diverting obviously certain things. But finally, when you're looking into the thing, the beauty of that reaction which I'm showing, you're producing, you're using the reducing power, and you're going to get back that reducing power also. That you can get back also in a reverse direction, you may be able to get back that reducing power also. That possibility is there. Okay. Therefore, in some of the cases, people always say that there's a possibility that it could be something to do with the storage of reducing power. Is it acting as a something to do with the storage of reducing power? Is it possible?
Okay. My second question is regarding the reducing power. So, under stress condition, glutathione is one of the another redox molecule under stress condition. A very significant level of the glutathione homeostasis need to be maintained because it's also ROS scavenging and antioxidant and all these. And glutamate is the key component amino acid of the glutathione as tripeptide glutathione. So that means the glutathione homeostasis is also must be disturbed. So what happened to the, because glutathione, if the glutathione is disturbed, then also it affecting a lot of other biochemical metabolism in the plants as well. So how that will happen, because that means glutathione need to be degraded to free up the more glutamate. So there is a lot of interlink in between.
No. No, I don't really agree with that part. We basically have to see, when obviously when you talk about glutathione, you're basically referring to ascorbate glutathione cycle, right? And obviously that has important role in this. But we have to weigh which is more important. Because if you basically look into ascorbate glutathione cycle, even ascorbate glutathione cycle is also having a role. That's another mechanism which is there to handle reducing power. But amongst two, which is cheaper? If you basically calculate that, this would be much cheaper when compared to that part.
Okay.
Okay. So this is protecting. There you're having an entire cycle that is operating, which is requiring multiple enzymes, which requires several other things. Here, as a single molecule, this is acting as so many important, it is handling so many things. I have not showed the things, it basically helps in scavenging radicals. Some of the radicals, it cannot handle superoxide anion radicals. We have shown that it can scavenge singlet oxygen species. Hydroxyl radicals can be handled by this particular thing. So this is something which is there. But only thing is, we have to see how many, we have to wait. Again, when nature has invented certain things, obviously it has reasons. Only thing is, we are trying to find out what are the reasons behind that. But amongst this, obviously, amongst ascorbate glutathione cycle, which is having several components in them, even ascorbate plays a very important role over there.
Yeah.
Right?
So that's what I'm saying. So glutathione is one of the key, playing key role as well. I'm not talking about ascorbate glutathione. I'm talking about gamma glutamyl cycle, which is part of the glutathione homeostasis, synthesis and degradation.
That is fine, but that is not going to be interfered much as far as this is concerned.
Okay.
You should not have much.
Yeah. But checking that level of glutathione under these conditions, and
we have done, some of our students did measure under stress conditions. I have not basically correlated that. After you said, maybe I'll try to look back into their data and try to correlate what could be the thing. Yes, sure.
Thank you.
Hi. Morning, Partha. Nice raising question and answering and completing the story. My question is regarding You mentioned that proline is acting as a competitive inhibitor to the oxygenase in place of oxygen. It will be repressing photorespiration, which is a major protective process against over-energization. So that is one aspect I will like you to clarify. And another is whether you have also studied these experiment with C4 plants. And one more question which regarded to, you mentioned that ratio between NADP and NAD. So you were saying that proline is being metabolized to rebalance the NADP for preventing the over-reduction. Did I get
No, I
it
said that yes, proline is basically being produced to oxidize reduced state of NAD to
produce it.
NADP to
Okay,
reduce.
so I thought
oxidized
that,
state,
okay.
yes.
So these two questions regarding that.
The first component, first
First
thing,
is that you are saying that it is suppressing the activity of oxygenase.
you are saying photorespiration is going to be influenced.
Which is a major protector.
No, that's fine. That is
okay.
that's perfectly okay. You are basically talking about photorespiration. See, I completely agree. So that's why anything which is basically being invented by the nature, I feel that everything has an important role to play. Because I have not looked in that particular way, so therefore I won't be really able to answer to that. But as far as photorespiration is concerned, of course it is having several other things. Let me not enter into that part. But I have not looked into that part. With regard to photorespiration, I never looked into that. I have to think about that.
Sir,
Yeah, we have tested that. No, we have tested that it's having almost similar kind of things. Almost similar kind of things. Yes, please,
Okay.
please.
sir, I have a quick question.
Yeah, fine. Sorry.
Actually, I work in the area of drought mitigating rhizobacteria. And one of the things we found, and we published it not very long ago, is that one of the ways by which these bacteria help in drought mitigation in Arabidopsis is by turning over proline. Instead of proline accumulation under, they help in maintaining a better phenotype. The drought tolerant phenotype is coming from high expression of both proline biosynthetic genes as well as catabolic genes, not just accumulation.
Exactly.
I just want to know what is your view on that.
I completely agree with that. I completely agree with that. See, there when we are talking about plant microbe interactions and doing the things, the situation will be different. That's why I'm saying that is one thing which we need to understand to a significant extent. That's what yesterday when they were talking about, two big professors talking about below ground thing, and especially talking with regard to root system. So that root dynamics is very important for us. So these are all the things which we need to understand. And of course, that's why I said when I was answering to his question, I was also talking about catabolism, not only anabolism. Both are equally important. It's not that it is going to remain at that particular stage. Whenever it is decreasing, it will automatically go down. We are also looking into that part. It does go down.
Okay. Hello, sir. This is Salendra from Botany Department, BHU. So my question may not be connected with your presentation because I missed the first half. I'm just trying to explore the things. Do we have the facility in India, or like if you know everywhere, like we can measure the redox state of the plastoquinone? And if we can do, how we can do that, the first thing. And the second thing is, like we talk about modifying the photosynthesis, exploring and doing some changes to maximize the productivity, all those kind of things. So I believe that the nature is the biggest engineer, and whatever the photosynthesis we have right now, this is the best one. So, and there is also the energetics things involved. Like if I'm changing something, it will cost something, just like a salary I get in the last, and then how I am spending. So how these things are going to give us the success, and I want to have your view on those things. Like.
So what you said, you already answered your own question. That is fine. Whatever you said is fine. But as long as the first thing is concerned, Professor Rajgopal is here. I think you should talk to him.
I have interacted with him. Like we'll be talking.
So you talk to him. I think he will be the best person at this stage where you will be able to perform those experiments.
Okay. Thank you.
I think now they are having best facilities. He's having best facilities.
Okay, sir. I will interact with
Yes.
you.
Limitation. Yeah.
Fine. Okay, fine.
We shall
Thank you so
conclude
much.
here. Thank you so much for the.
One question, sir. Very small query.
Yeah.
I really like the talk that you learned so many things about proline and its role. So just very small query about it. Like the way you said that proline is also binding to the site where oxygen is also binding. So are the kind of interactions or bonds are similar between oxygen and proline? Or there's any difference or the conditions are different?
I don't want to make any comments because it's only preliminary thing. I don't want to jump to any conclusions based on my preliminary things. But the possibility is there. There could be. I don't know how it is going to happen because let me not make any big claim on that. Huh? Right.
Thank you.
Thank you so much, sir.
Thank you, Professor Parthasarathi, for an engaging lecture. Thank you very much. I invite Dr. Deepu Matthew, chair of the session, to present the memento to Professor P. Parthasarathi as a token of our love. Sir, please. Thank you, sir.
Good morning, all of you. With the permission of the chair, I am introducing the second speaker of the session, Dr. Ashwini Parikh. Dr. Ashwini Parikh is the Executive Director of National Agri-Food Biotechnology Institute, Mohali, Punjab, where the 5th International Plant Physiology Congress is being organized during 6 to 9 January 2023 in association with ISPP. Dr. Ashwini is also serving as the Chief Executive Officer, Center of Innovative and Applied Bioprocessing, Adjunct Professor at RCB and University of Western Australia, and Professor at Jawaharlal Nehru University, New Delhi. Dr. Ashwini's thrust areas include plant physiology, plant molecular biology, rice genetic engineering, and nutritional enhancement. He has played a pivotal role towards genome-wide analysis of stress-related gene families in Arabidopsis and rice. Some of his prestigious awards include R.D. Asana Memorial Award, Tata Innovation Fellowship, J.J. Chinoy Gold Medal Award, INSA Royal Society London Award for Exchange Visitors Fellowship, Boyce Cast Award by Ministry of Science and Technology. And he's also the Chief and Associate Editor of a number of national and international journals. And I warmly welcome you, sir, for the talk. Thank you, sir.
Am I audible? Okay. Very good morning to all of you. I think I would like to put on record my sincere thanks to Josh Puthur, good friend of mine, who has kindly invited me over here to share some of the ideas, some of the research which we are conducting at Jawaharlal Nehru University. My purpose of coming over here is not to tell you something new, some science about it, but I will give you an idea why am I here. I'm here because of two reasons. Number one reason, I always enjoy talking to students because they are the future of science in India. And I would like to give you a message from my talk over here that the person whom you are seeing in the picture is, I think all of you know about it, we are celebrating his 90th birthday. Why am I showing his picture is something very, very important. This person at the age of 90 is full of energy, is full of enthusiasm, is full of passion, is full of hard work, is full of all good things on the earth. And that is the reason he is also known as Mr. Photosynthesis. Mr. Photosynthesis because he has done so much work in the area of photosynthesis and is still continuing his work in the area of science. The message I want to give all of the students that you should be passionate about what you are doing, and once you start dreaming about it, you realize that more you are learning, less you would know about the subject. And that's the message I want to give you from this picture. This picture was taken in a small village in Rajasthan, which is my hometown, Sambar Lake, which is the largest natural saltwater lake, which you can see at the background also. So he kept on visiting us before COVID days at Jawaharlal Nehru University, where he was teaching our master students and helping some of us in doing wonderful research. So my lecture today is actually dedicated to Professor Govindji, who is the man of passion, man of science, man of photosynthesis. And this picture which we are talking about here is, I took him to my village. He was so much fascinated about the work we were doing on some of the zero halophyte that he decided to come with us. Neeta is also sitting here. She was also accompanying her. My lab at JNU, my lab at ICGD, we hired a bus, almost 40 people. We went there. We stayed in the lake resort over there, and we enjoyed the typical Rajasthani food over there, dal bati churma, if all of you know about it. And we started working on one of the zero halophyte, which is shown over here. I will not give you the details of this work, but I would also like to tell you that he's still continuing with his passion about his hard work and the work. And all of you have heard that behind every successful man, there is a woman. And literally you can see behind this man, you have a wonderful woman. They're supporting each other, they're living together, they are putting hand in hand together in hard time, good time, bad time. And I spoke to him this morning. Madam Govindji had a fall in the morning, and she is recovering from that fall. So I wish him good success, healthy life, and productive life to both of them. And my lecture today is dedicated to them. Okay, so I'm taking care of this institution, which is NABI institution. All of you know about it by name, National Agri-Food Biotechnology Institute, which is under the Department of Biotechnology. The vision of the institute is to ensure that whatever we are eating is nutritionally balanced and enough food is available for all of us. So I take this opportunity to invite you to Mohali campus in NABI, and this is another institution, Center for Innovative and Applied Bioprocessing, which is actually engaged in doing bioprocessing, secondary agriculture, and bioeconomy. And they want both these institutions. Some of my friends sitting over here, Om Prakash, they have visited us. They are wonderful institutions, so I take this opportunity to invite all of you to visit us, and I'm going to give you a slide at the end of it where we are inviting you to the next conference, international conference, which we are organizing from 6th to 9th of January. And this is going to be the meeting, wonderful meeting under the aegis of Indian Society of Plant Physiology, Plant Tissue Culture of Association of India, American Society of Plant Biology, and we got a message yesterday that Crop Science Society of America is also willing to help us in that process. So I would like to tell you that what we are discussing over here for the last 2 days, and the lecture before me was a wonderful lecture by Professor Parthasarathi, who's been a wonderful teacher, and we all keep on learning whenever he is presenting. So my talk is going to talk about giving you some ideas what we are doing and ensuring that whatever resources we have in India, in Asian countries especially, we are able to give enough food to the people. And the reason I'm saying is very, very important because India is a special country where we don't have to feed 27 states by the number of mouths we have to feed, but 27 countries at the population of many of the states in our country is actually equivalent to population of many of the countries of the world. So we have to feed 27 countries. And most of us also agree that in India, the concept of nutritional or balanced food is not known to us. We primarily depend on rice and wheat as the staple crop, the carbohydrate crop. Many of us do not have access to vegetables, fruits, and all that. So I think food and nutrition security has a special reference when it comes to feeding the population in our country. So my talk is going to ensure that we have some ideas, some hope towards ensuring this food and nutrition security in our country. So I spoke about my dream just now when I spoke about that I belong to a small village in Rajasthan, which is a natural saltwater lake. It is largest in Asia, where the salt is being produced commercially since ages. As you can see some of the heaps, the laser pointer doesn't work over here. But the white thing, if you see on that, the salt is being produced commercially since ages. They take out the water, they let it dry out, water evaporate, and they collect the salt from it. All commercial varieties like, Surya Namak, Rajdhani Namak are actually being produced over there. So that's my hometown. We had a small farm over here. One fine morning when I was doing graduation at Delhi University, my father called me from my village. By the time he had retired, and he said that the well which we were using for irrigation in our field has turned saline because it got connected to the lake. Monsoons are becoming unpredictable for the last 1 or 2 decades, and farmers are extracting more and more water from ground-based resources. So if you can assume a situation when you have a lake full of salt water and the neighboring villages are drawing more and more water, the water from the lake is going to percolate down to the well. And when you use that water for irrigation, you actually cannot grow anything. So that day I decided that I will have dream, and that's the second message I would like to give you, that those students who are interested in doing good research, they should always keep a dream in mind. They should always remember that they have a dream in mind. They should always visualize themselves that where do they want to be 5 years from now, 10 years from now, because if you have a dream, you can try to fulfill that dream. So I had a dream, which I'm showing over here. Those of you who are interested in it, there is a video available on YouTube, Alliance for Science. You can just Google around. That will tell you my village and the story I have. But when I took my students over there, when I joined Jawaharlal Nehru University, and my master's and PhD program was also in salinity, we were actually very happy to see that in this white desert of salt, there's still some green color. Professor Parthasarathi spoke about photosynthesis, the green color. We were very happy to see that photosynthesis and salinity are not mutually exclusive. Some plants can do photosynthesis under extreme conditions of salinity and high temperature and drought. So we started exploring some of these micro below ground resources and some plants. I will not present the story. I will present stories which are relevant to today's talk, as we are talking about Professor Govindji over here. I will present some short stories. Nowadays people are seeing a lot of serials on TV. So they have season 1, episode 1, episode 2, episode. So I'll give you some episode which will make you happy that yes, the story is coming up in a big way. Before I do that, I would like to thank all my funding organizations, some of the students who are, Anita is sitting over here as well, some of my collaborators, nationally, internationally. So I'm just speaking on behalf of my collaborator, students, and the funding agency. A big thank you to all of them who is enabling me to stand before you and share some of the pictures. We work on rice because I believe that rice is a crop which is meant for poorest in the country, and rice is a crop which is meant for richest in the country. So even if you do small incremental improvement in enhancing the yield or the quality improvement in rice, it is going to play a very important role in an economy and Indian sustainability as far as agriculture is concerned. And rice is also a model crop. It has a lot of diversity we talk about. International Rice Research Institute, which is in Manila, iri.org, if you can log on and look at it. Out of 1,73,322 rice genotype, more than half are actually coming from India. And I'm going to pick up a story which is very, very relevant to this particular state, God's own country. I always call Josa. I enjoy coming over here. Kerala we are talking about. In Kerala and many of the coastal countries, they have exploited the rice diversity, and I'm going to give you a short story on that. But before that, I tell you that why did we pick up salinity to work on, and what did we learn from that? The take-home message for all the students, the first take-home message is over here. The stress response, specifically salinity and drought, is highly, highly complex. Why do we call it complex? Because whatever parameter, whatever measures you take, it tells us that it is not controlled by one gene, it is controlled by multiple genes, and there is a crosstalk, and a lot of networking goes on as far as gene network is concerned. Number one is that the stress response is cell and tissue specific. What I mean by that, if you take root and shoot, the response is going to be different. Within the root, if you take apical meristem cell or you take marginal cell, the response is totally different. Second thing, it is a dose specific. If you are giving 50 millimolar NaCl, sometimes it is inducive as well as a mild stress. And if you go for 200 millimolar NaCl or 300 millimolar NaCl, it is lethal, as is true with many of the stresses. It is stage specific as well. If you talk about rice, the seedling stage seems to be the most sensitive stage as far as salinity is concerned. Then comes a reproductive stage. In between, the vegetative stage is quite sturdy as far as salinity is concerned. That brings in a lot of complexity as well. Early and the late response. If you look at the response of the plant after, say, 5 minutes of stress, 1 hour of stress, 2 hours of stress, the response is totally different as compared to you see the response after, say, 48 hours, 96 hours, 10 days after that. Because the late response is often associated with the genetic basis, and the early response is associated with osmoregulation, as Professor Parthasarathi also spoke about. The genotype specific, I spoke about diversity in rice. The response, if you see in IR64, which is sensitive, vis-a-vis Pokali, which is a farmer selection from this area, coastal area, is totally different. And we also discovered that it is also influenced by the diurnal rhythm, because photosynthesis goes on, compatible solute goes on, and plants tend to actually adjust. The fine adjustment, the micromanagement goes on as far as the induction of gene is concerned. So taken together,
The message is that the response is quite complex, and the message for the student is that if they're trying to study the salinity or the drought response, they have to take care of all the parameters, meaning thereby that if you're harvesting a sample at 10 a.m. in the morning, the next sample, if they're looking at the cycle, at the duration, they should harvest it at 10 a.m. because if you harvest the sample at 5 p.m., the response may be affected by the diurnal rhythm as well, which is not yet reported many times in literature. Of course, when you look at the molecular response, not our own laboratory, but other laboratory around the globe, have been working towards understanding the response of plant toward these abiotic stresses, especially salinity stress, and they have again shown that a lot of molecular machinery are associated with that. We just learned about osmoregulation, but besides that, you have detoxification enzyme, SODs we talk about. There are specific ion transporter. I'm sure Shargi must have spoken about transcription factor, a protein kinase, a signaling compound, and the response is actually very, very complex when it comes to salinity stress. As far as signaling is also concerned, you can see that several pathways have been discovered. Most important one is the SOS pathway, SOS3, SOS2, SOS1. I will not go into detail of that, but giving us an idea that within a cell, it is a coordinated and orchestrated action of all these genes, enzymes, and the protein which is making a plant tolerant and sensitive. So in our laboratory at Jawaharlal Nehru University School of Life Sciences, we are actually using two kinds of approaches. In India, we are not permitting the consumption of genetically modified food, though we have several products available from our own country. 2 days ago, in a major historic decision, the Ministry of Environment and Forest has cleared the genetically modified brassica, and we hope such decisions are going to encourage more and more research. But practically speaking, not even a single crop beside this brassica which you're talking about, mustard you're talking about, had been permitted to consume in India. So we have to go for other methods. So the two kind of methods which we are using in our laboratory are reverse genetic, where we pick up some specific alleles from the tolerant genotype, try to characterize them, try to move in a translational way. And I'm going to show you a story of one of the member, HK3B, which has been talked about from our laboratory, which has been patented in several countries. Almost 24 of such genes have been characterized by functional genomics tool. Almost 12 have been patented in different countries from our laboratory. And the other approach which we are using is the forward genetics, which is a very, very powerful approach, where we create the mutant, and then when we find the mutant which is able to tolerate salinity stress, we use all kind of molecular tool to understand what has been changed in that particular mutant. So these kind of tools go hand in hand, which is giving us deep insight into understanding how salinity response is controlled in crop plants such as rice. I will present a couple of short stories to give you an idea. I will not go into detail of it. Those of students who want to know more about it, I will be available after the discussion, after the talk as well. So one of the approach which we are using in a laboratory is a very powerful approach. I call this powerful approach because for a molecular biologist, if you have contrasting genotype, one being sensitive, which is IR64 over here, which is very, very high yielding, and another one is tolerant, which is from the local area, coastal area, such as Kerala, which is salinity tolerant. These kind of contrasting genotype, they are gold mine for a molecular biologist who's interested in knowing the details of that. Why? Because one is sensitive, one is tolerant, both are rice, and rice genome is known. Rice is the model crop. You have a lot of molecular tools available. You can actually go deep into the physiology, into the biochemistry, into the microscopy, into the anatomy, into the molecular biology, and try to understand why Pokali is able to tolerate stress and IR64 is or not. And that is exactly what we did. We used the tools of omics. On one hand, we looked at proteomics, metabolomics, ionomics, transcriptomics, various platforms which we used over the last 2 decades. I'm just showing in one slide and published a lot of paper on that. I will skip the details of that. But this has given us a lot of understanding that why, and we're focusing on seedling stage only because there's so much to know at the reproductive stage, and the physiology and biochemistry is totally different from a seedling in the reproductive stage. We are focusing on seedling stage to go deeper and deeper into that. But this has given us a lot of confidence in proposing some of the fundamental aspects about the salinity tolerance. And I'm going to show you one of the fundamental aspects in a minute from now. The work from our laboratory on Pokali has been well recognized by various newspapers from your local language as well. Paddy in Brine, this is a very wonderful article which was given by one of the reporter, where actually you can see, I'm not able to use the pointer, where you can see the rice is actually growing in saline water, which is the sea water. What else you need? That means you already have a plant which is able to give you productivity under saline condition. But the problem is that the yield is too poor for the farmers to be able to use it for an economic model. But yes, nonetheless, as I said, for a molecular biologist, this is the gold mine. And our laboratory has been interested in understanding why some genes in Pokali are different from those in IR64. For technical term, we call it allele mining. For every gene, you have multiple copies. Like Mendel did, you have a wrinkled gene, you have a smooth gene for P. Similarly, we have alleles for different important genes. So what we are trying to understand is that what is specific in Pokali about some of the alleles of important gene. And I'm going to give you one or two examples to tell you that allele from Pokali is different from allele in IR64. And this is what we are doing. So these are several success stories which have been published from our work in local newspaper. I just picked up some of the news cutting from the local newspaper over here. But I'm just going to share one slide out of the whole work. When you look at the transcriptome, proteome, and metabolome between IR64 and Pokali, you realize that the two plants behave very, very differently. And one of the differences I'm showing over here is in the form of the transcript abundance for the benefit of students. I actually wanted to show you, we have done real-time qRT-PCR, all those things we have done, but I think I still like the way this traditional way of looking at the transcript abundance by using northern blotting is concerned. Shown over here on the left side is IR64, on the right side is Pokali, and C means control, 10 minutes, 20 minutes, 30 minutes stress, 24 hours, 48 and 72 hours, and then densitometric analysis shown over here. The message I want to give you from this particular slide is that even if you look at the genes which are belonging to the kinases, transporters, and all that, you can see that each gene has a unique pattern of accumulation when it comes to transcript abundance, right? And you can see each gene has a dancing pattern, and dancing pattern of a gene in IR64 is different from that of the Pokali. So the message I want to give you for the student is that it is a coordinated and orchestrated, I will use the word again, coordinated and orchestrated action of all these proteins which are encoded by these transcripts and which actually determine the metabolite, metabolome, and hence the biochemistry of that makes Pokali tolerant as compared to IR64. So the take-home message from this particular part of the talk is that what we have learned by using transcriptomics tool, proteomics tool, and metabolomics tool is that Pokali is able to tolerate stress because it is ready in anticipation. The transcript level of some of the key genes which are otherwise inducible in IR64 are constitutively kept abundantly high under non-stress condition. I would like to give you a very crude example over here, which, suppose if you are sitting over here and we know that somebody is going to come with gun to kill all of us. There are two situations now. All of us carry a gun and shoot the person down. This is one situation. We are ready in anticipation. And the second situation is that we don't even know who is coming, and when the person comes, he or she shoots us out. There's a well-preparedness of the system which enables Pokali to maintain higher transcript, higher protein, higher metabolite. Of course, it is an energy drain, and that is the reason yield in Pokali is low as compared to IR64. But this gives us survival strategy, which is very, very important. And if we can understand the survival strategies by analyzing some of the genes, some of the proteins, we can actually make IR64 tolerant. And this is what exactly I'm going to show you next couple of slides. So the take-home message is that Pokali is tolerant because it is happy, and people who are working with Arabidopsis and Thalangella, Brassica nigra, Brassica juncea, this seems to be the same uniform mechanism by understanding these things. So this is something fundamental which we have discovered, that plant tolerate because they are ready in anticipation. Out of several genes which we have characterized, one of the genes which I presented last time, so I'm just showing you one slide. I came here 3 years ago. Jose invited me. I shared the whole story of this osmosensor which we have discovered from rice. This particular protein is a membrane-bind protein. We have worked out the mechanism, and this has been patented in several countries now. But we have found out not only the protein which is responsible for sensing the osmotic stress, but the whole osmosensing cascade down to the several genes which are transcription factor we have been worked out. So I will skip all those details for the benefit of time. But the functional genomics tools are giving us wonderful insight that how these members of two-component system, which are hybrid type histidine kinase, are playing a role as far as sensing and sending the signal to transcription factor in the nucleus is concerned. And this work has been translated. We got all the patents in Europe, USA. The JNU has given this technology to an industry. And what I learned from industry that they are very happy with the product. The plant which we have generated, which we call strong rice, stress-tolerant rice of next generation, are very, very tolerant to both salinity and drought. Almost 30% higher yield we have been able to generate. And the company has also tested now. They are also very happy with the product. So it's very satisfying to see that your baby is also happy in somebody else's lab as well. Which is, as scientists, I think it is very, very relaxing, very satisfying to see that industry is giving good feedback. But we were fortunate enough that this work was picked up for an award by the president. We were invited to Rashtrapati Bhavan for this Best Technology Award a couple of years ago. But this has given us confidence that understanding nature, understanding how nature is actually solving the problem, can actually give you a lot of good insight. And if we can use that insight to develop something productive, you can actually move in a translation mode. That's the message I want to give. So coming to this part of the talk, when I'm talking about some of the episodes I'm talking about, very briefly I will touch upon that. All of you know that salinity is a complex trait. Several QTLs have been identified. One of the QTL is Saltol QTL, which is on chromosome number 1, which has almost 900 MB of gene. So we got interested in this QTL because this QTL actually contributes to almost 43% of salinity tolerance at the seedling stage. We did a lot of investigation. To cut the story short, we realized that a lot of genes are present in this particular region. We focused on some transcription factors, some signaling compound, and I'll give you one particular story where Professor Govindji has been associated with that, and that is related to intermediate filament. So what we did, that we actually looked at this particular gene, which is intermediate filament. So I would like to draw your attention towards the fact that we made several libraries, cDNA libraries, subtractive library. One of the most abundant gene which we found is the OSIF, intermediate filament, which is the cytoskeleton gene. So just to give you a brief on that, the plant cytoskeleton, microtubules have been reported, and they have been report of microfilament, but in plants, intermediate filaments have not been reported. So our lab is the first laboratory which has given this fundamental contribution to the plant science, that plants do have intermediate filament, and they have very much role not only in giving structural architecture to the plant, but they are also important for abiotic stress tolerance indirectly as well. And I'm going to give you evidence for that purpose. If you look at this transcript abundance, which we analyzed by real-time PCR, the white bar shows the abundance in IR64, which is sensitive. And the black bar shows abundance of transcript in Pokali. And you can see again under non-stress condition, which is on the left side, CON, Pokali shows higher transcript abundance. And this gene is inducible by mannitol, which is a kind of osmotic stress, high temperature, abscisic acid, heavy metal, methyl viologen. So this is a multi-stress responsive gene. For those who are working on abiotic stress, they will agree with me that the need as on today is to have the genes which are responsive to multiple stresses, so that we can engineer the plant for more than one stress together. We did a lot of microscopy, genetic evidence and all that, but we realized that this is interacting with metallothionein. We looked at subcellular localization. We expressed them in bacteria. And to show over here in the last plane, if you see, the bacteria which are accumulating this intermediate filament are actually able to tolerate salinity and drought stress better in the form of mannitol. In yeast also we did the same thing. In tobacco also we found the same thing. So from evolutionary diverse organism, from bacteria to yeast to Arabidopsis, finally, to cut the story short, we looked at the transgenic plant which are altered in the expression of them. These are the targeted mutant which we created. So when I say OIEF2, OIEF1, these are the plant which are overexpressing this gene. That means they are having higher abundance of this gene. When I say KDIF, the knockdown gene, the RNA mechanism where the level of this protein are very low. And you can see very clearly, if you look at the expression, the phenotype of the plant, which is shown in the lower panel, the plant which are overexpressing this intermediate filament, they're performing better under non-stress condition as compared to wild type. And on the left side, you have the knockdown plant. And what happens in the stress condition, very, very dramatically, very clearly you can see that yes, the overexpressing plant are performing better. We did a lot of investigation, and to cut the story short again, Govindji got interested in looking at photosystem 1, photosystem 2. What I learned from him, that yes, the plant which are having higher abundance of intermediate filament, their photosynthetic apparatus is very much protected even under salinity stress condition. It is like, you have more pillars, more iron bar in the building, in the chloroplast, and the chloroplast is now able to sustain more of osmotic stress. That's the message I got from this particular work. But if I show you the yield of these particular plant, which are shown on the right side, middle one is the wild type, on the right side you have the overexpressing plant, under salinity, under drought, again these plants are showing very, very important. So this understanding which we got based on expression pattern, because Pokali was showing higher abundance of this transcript, we picked up this gene, we picked up this allele, overexpressed in IR64, and we got a product. So this has been patented as well now. The second story, very briefly, I will tell you about, Professor Parthasarathi spoke about the molecular chaperone. So this particular chaperone is a very interesting chaperone, which we call cyclophilin, and this is also known as cis-transpeptidyl prolyl isomerase, because actually this is converting the trans form of a protein into cis form that is actually affected under stress condition, be it high temperature, be it saline. This particular protein actually twists the protein just before proline so that the protein takes proper shape. It's a kind of molecular chaperone. A lot of work which we have done, and to cut the story short again, this gene is also inducible by several stresses, again expressed in bacteria, in yeast, in tobacco, and again in rice. What we learned over here, that overexpression of the cyclophilin, we worked out the mechanism also. If you look at the overexpressing plant, which are the second and the third number in the left, wild type is shown on the left side, you have again the higher yield under drought and salinity stress condition. Again, this is an example where we learned that Pokali is having higher abundance of this transcript, higher abundance of this protein. We hooked up 35S promoter and picked up the allele from Pokali expressed in IR64. Now IR64 is actually showing you tolerance. Why am I showing this small epitopes? I will tell you at the end of the story. I will skip the other gene which we worked about is SOS member, SOS1, SOS2, SOS3. This is the pathway which Zhangqiang Zhu's group in China discovered. SOS3 is a calcium sensor, SOS2 is a serine protein kinase, and SOS1 is a sodium proton antiporter. So the cascade of SOS3, SOS2, and SOS1, it maintains the iron homeostasis. So we have worked on all these three genes. I'm showing you a story on, this is the pathway which you are showing about. I'm going to show you the story on SOS2 as well. If you see the overexpressing plant, which are shown as OE19 and OE15, second and third panel, under drought condition and under salinity stress condition, again these plants, again the gene, the allele has been taken from Pokali and overexpressed in IR64. We looked at several sodium accumulation and all that. But the take-home message from this part is that, we have a plant in front of us where nature has evolved Pokali. Farmers have been cultivating this particular rice since ages, and slowly it has evolved. The mutations have been taking place in different genes of this particular plant, and if we understand the slow molecular evolution, now the tools of genome editing are available, as also mentioned by Professor Parthasarathi, that we can use this precise gene editing tool to understand what are the differences between the alleles of IR64 and Pokali, and if we can work out those precise base pair mutations, which is possible through gene editing, we can actually control the plant as far as the salinity response is concerned. So at least in countries where salinity and drought are the major issues, we can have confidence that yes, in years to come, when we are going to be 9 billion, gene editing technology and genetic modification technology are going to speed up the process of delivering salinity and drought tolerant rice in year to come. So we have several such stories. I just wanted to give you a message that yes, the response is quite complex, but if you try to learn it from nature, you can make use of the basic information and the learning from nature and convert it into translation mode, and you can develop product. But some messages I would like to give you for that. Gene pyramiding for sustain, as I showed, overexpression of SOS2 gave you tolerance, overexpression of intermediate filament. So I think time has come when we talk about gene pyramiding, and my laboratory has actually been working on gene pyramiding. I did not present the work over there, but when you pyramid the genes such as SOS2 and HK on SOS2 and SOS1, they give you actually synergistic effect. So gene pyramiding is going to be very useful. Do we have any solution for patchy patchy salinity. This is a new concept which is coming up. Within a farmer field, if you go, somewhere the salinity level is too high, somewhere the salinity level is not too high. So are these technologies which we are developing, new varieties, salinity tolerant variety, are they going to take care of this patchy salinity? Yes, we need to devise plant which are not having any energy drain under non-stress conditions, so that the plant which are not facing salinity will be able to give you even higher yield. So those kind of consideration we have to see very, very carefully. Need to test for the combined stress and sequential stresses. We are also moving in that direction. Several publications we have from our lab where we are trying to understand that what is the response of a plant when it comes to combination of stresses. Salinity and drought are often coming together. Drought and high temperature is often coming together. Post-emergence drought is often coming together. So I think time has come when we try to, make our life more complex, more difficult by doing this combination studies. How can sustainable agriculture that is protective, we have to look at the biodiversity also. We are talking more about that. I think the example which I just showed you is a wonderful example where we can learn from nature and we can appreciate the biodiversity and maintain in a very good way. How can societal acceptance of current gene editing technology be achieved? This is something which I would like to spend 1 minute over here. We have already burnt our hand when it comes to commercialization and social acceptance of genetically modified food is concerned. BT brinjal has been approved in our neighboring country, Bangladesh, and 2 days ago, even Philippines approved BT brinjal. I think time has come now. I'm very happy that this mustard, GM mustard has been approved. So I think each one of you who is practicing science should act as an ambassador of the technology and believe in the technology, have faith in the technology, that yes, what we are doing over here is not against the nature. The recombination is already taking place. The gene mixing is already taking place. Agrobacterium is a natural genetic engineer. So if you go in totality, we are not doing anything unnatural. So when it comes to gene editing, let us be proactive, let us be receptive to the technology, because if we do not accept these technologies with open arm, perhaps we'll be lagging behind. And these products cannot be generated over time. If we have to ensure that 10 years from now, the food should be available when we are going to 9 billion, we have to start preparing as on today. We have to protect our IP as on today. We have to develop the product as on today, because these products cannot be generated overnight. That's the message I want to give all of you. Can cutting-edge fundamental societal science be integrated to address supply and demand? I think we need to address these challenges as well when it comes to the science, global food system we're talking about. What cross-disciplinary actions are required as a personal? I'm using the word personal. All of us can act as an ambassador, public, and planetary level. We have to go at the planetary level when we say that we have only one Mother Earth which can sustain ourselves. No other planet can do that to ensure successful outcomes. I think when we are doing science, we have to be very, very clear. We have to learn from nature, and we have to continue striving hard. As I said, Professor Govindji at the age of 90 also, he's still asking questions, still working with many of us in many universities. So hats off to him. So this is the invitation I would like to give you. Please mark your calendar. This is going to be the event in the country where I can tell you the Chief Editor of Plant Physiology, Chief Editor of Plant Cell, Chief Editor of Plant Biotechnology, General Food and Energy Security. A lot of good speakers are coming, and the registration is going to close by, I think, Saturday evening. I'm going to close that because we have enough number. My auditorium at Nabi, Dr. Om Prakash Yasin, cannot accommodate more than 450. So I have to stop the registration by Saturday evening. So those students who are interested in listening to good science, please, I welcome you all. This is going to be the event in the country. The conference is International Conference on Food and Nutritional Security. This will be held under the aegis of Indian Society of Plant Physiology. I'm thankful to that. Plant Tissue Culture Association of India. This will be jointly handled by NABI, SIAP, which are in Mohali, ICGEB, which is in New Delhi, National Institute of Plant Biotechnology, which is in New Delhi, American Society of Plant Biology, Crop Science Society of America. So a lot of good things are going to happen. Please, I would like to request you to make down note of this date. This is IFANS 2023, and I'll be very happy to host you at my Mohali wonderful campus, beautiful campus. And with this, I would like to thank once again Jose for this opportunity. Thank you very much for your patient listening.
Thank you so much, sir. It was indeed a energy-packed as well as information-packed talk, a very nice talk. You have started with an inspiring picture of Govindji, actually working in the field, and also introduced the diversity of rice in India, and with a special reference to the Pokali from Kerala. And also, you gave a glimpse on the molecular mechanisms, the intrinsic complexity of the molecular mechanisms involved in the stress tolerance. And also, you have stated and shown that the coordinated and orchestrated action of the spectrum of genes is actually leading to the resistance. And you have briefly discussed which are all the major works you are involved in. For example, the identification of the OSHK3B protein, which is a very important membrane protein, which led to the development of the variety strong, stress tolerant rise of the next generation. And secondly, the identification of the QTL maltol and the associated genes, especially the intermediate filament gene or the OSIF gene, and how you have transferred it to the other crops, even to the bacterium and all, and how that is actually offering the tolerance. Subsequently, you have again discussed the molecular chaperone, OSCYP2P gene, and also the complex networks, the SOS1, 2, and 3, and how you are progressing in that. So thank you so much, sir. It was a great talk. We all are benefited. And questions?
Thank you very much for excellent talk. Always, I enjoy your talk. And more than the talk, I enjoyed your enthusiasm in that one, the way you speak in that. So, one comment first and then a question. The comment is about the genetically engineered crops you made about. I don't know why we genetically engineered sounds like a negative connotation. Why we call this genetically engineered? Why don't we call them genetically enhanced plant? Because we are basically enhancing the expression and changing those, right? So why should we calling negatively in a way? We should change that mindset.
Yes.
Second, my question is that you have all this excellent work and all the genes that you identified in Pokali for salt tolerance. And you are characterizing those individual genes and overexpressing and doing this. Why not we go the way we did with the sub-1 rice genes, right? You have all those genes. Why don't we introgress those using the marker-assisted breeding? That will be going faster in the field. And some of those genes may be closely linked, could be used with the same marker, and you can basically stack those that way.
Okay, so I'll address the first question. I think I also agree with you. We should stop calling them genetically engineered. Genetically enhanced is the right term. And this is what exactly when I addressed some of the reporters in New Delhi 2 weeks ago, when gene editing a memorandum came on March 30th. This is exactly what I said. Thank you very much for reminding me. But I think, as I said, all of us have to act as an ambassador that this technology is going to see the day of the light, and it cannot do it unless we welcome it by open arm. The second question, which Dr. Om asked, wonderful question, that why can't we actually pull in the two QTL? One is the sub-1 QTL, which is for submergence tolerance, and the salt-told QTL. So there are two reasons. I'm not doing it because my lab is not having field. When I was working in JNU, we did not have access to field condition. So what I learned from Dr. A.K. Singh, they already have a big project where they're trying to introgress this salt-told QTL into sub-1 Swarna. And what I spoke to him almost 2 months ago, Dr. A.K. Singh from IARI, that they are getting good success in that. So I think science is full of different tools. Each one of us should keep on trying different tools, assuming and expecting that success is lying somewhere nearby. The QTLs are important, the genes are important. Whichever means we can do, I think we should continue striving and moving towards the goal. My lab is doing that in my own way, but yes, IRI, since they have a different toolboxes, they are doing at their own level. Thank you very much.
Yes, Ashwini, I just have one comment about your last comment, whatever you made. Acting as ambassadors for promoting GM technology. I have my reservations about this. Now, can you give me an example where we have clearly demonstrated that it is superior to hybrid? For example, you are talking about GM mustard. Have you demonstrated anywhere that hybrid, whatever GM you're talking about, is it superior to hybrid mustard? Did you ever compare that?
Okay, the question is that do we have enough scientific support, where we can say that the product from genetic modification, genetic engineering is superior to other varieties which are available. So this GM mustard specifically you talked about is basically towards developing a system for ensuring the male sterility so that they can cross it and they can actually do have the Barnes and bar star system so that they can make the hybrid. And of course, this is herbicide resistance also. So the yield is not the target over here, but when you use these kind of tools, when you use herbicide tolerance and you use Barnes bar star system to do precise I would say pollination, you can bring in pollen from a particular variety and put it onto the stigma of that. And one of them is actually sterile Barnes-Barsa system. We can actually control it very well. The yield enhancement is not because of Barnes and bar star, but the varieties which you can cross using this particular system. That is my understanding. But yes, when it comes to salinity and drought tolerance, the rice which we have generated, I did not show you some of that which we have done it in the microplot level, and now we are doing at NABI also. Yes, these single gene changes are actually bringing dramatic effect when compared to the traditional wild type IR64 when it comes to the yield under stress condition. For salinity tolerance, for drought tolerance, I can be assured, I can give you in writing, yes, these are superior to the traditional IR64.
So sorry for interfering. Maybe we have the third speaker already.
What would be the long-term effect?
No, especially the Indian mustard looking at, I think it's going to have negative impact, especially as long as the environment is concerned. I mean, my major concern as a professor of environment is the negative impact, especially excessive use of herbicides. And that's negative.
But as it is, we are using herbicide. I mean,
we have to, that's what I'm saying now. Let's not try to promote that further.
No, but by using these herbicide tolerant plant, you are going to reduce the concentration of herbicide in the field condition.
Yeah. Sir, we will have the discussion maybe in the lunch time. And we have the speaker for the concluding lecture.
Hello,
Dr. Christine H. Foyer. Yeah.
Thank you, Professor Ashwini Parikh, for such an engaging and informative talk. Now, I would like to welcome Dr. Sumesh KV, co-chair of the session, to present the memento to Professor Parikh as a token of our gratitude. Sir, please.
It's good to see Christine online.
Thank you, sir.
Good morning, madam. A warm welcome to our international seminar. Friends, we have a great speaker here, an authority of plant physiology in the globe. Dr. Christine H. Foyer is a professor of plant sciences in the University of Birmingham. She will be talking to us on the roles of reactive oxygen species in plant growth and defense. And she received her PhD in 1977 from the Department of Biochemistry, King's College, University of London. And she is an expert in plant metabolism and its regulation under the optimal and stress conditions. And she is a chair of so many scientific societies, and she received very many awards. I am not going to detail one by one. And she has published more than 400 papers in highly rated journals, and the number of citations are 81,652. That's what printed here. And today morning I have Googled, and within this span, now it has crossed 82,000. So, madam, we are all highly fortunate to have you here. And thank you so much for joining us. And without wasting more time, I am inviting you to deliver the lecture. Thank you so much.
Thank you so much. I hope that you can hear me without problem. Is the sound okay? I hope so.
Yeah, we can hear you.
Yeah, thank you, thank you. I am so very sorry that I can't be with you in person today. It would have been a great pleasure, as always, to be in India and to certainly be with you and your colleagues. It was a great pleasure to see my dear friend and colleague, Professor Ashwani Parekh, deliver the last lecture. And also to see in the audience Professor Om Prakash and many other colleagues. So at least I can talk to you remotely, even though I cannot be there in person. So today I'm going to talk to you about a topic that has interested me for all of my career. I started looking at the roles of reactive oxygen species and the systems that are important in protecting against reactive oxygen species during my PhD. And since that time, when the field was very small, there has been an explosion of information and knowledge. So from very humble beginnings, this topic is still continuing to expand and to be important in many, many aspects of plant biology. So just to start at the very beginning, the world that I work in really involves all the systems in the plant cell that produce hydrogen peroxide. So we have, for example, the chloroplasts here, which generate different forms of reactive oxygen species, singlet oxygen at photosystem 2, and superoxide and hydrogen peroxide at photosystem 1. And also, hydrogen peroxide is generated as a result of the process of photorespiration, which transports glycolate out of the chloroplast to produce hydrogen peroxide in very large amounts in C3 plants within the peroxisomes. We also have the mitochondria, which produce large amounts of reactive oxygen species by virtue of the electron transport chain. There are many, many sources in addition, enzymes, etc., that produce hydrogen peroxide within the intracellular environment. What's also particularly important in plant responses to stress are the enzymes which are involved in the production of reactive oxygen species on the plasma membrane, particularly the family of respiratory burst oxidase homologs, or NADPH oxidases, excuse me, whose job it is to produce superoxide and hydrogen peroxide in response to any kind of physical or chemical change. In addition, there are other enzymes within the apiplast cell wall compartment which produce hydrogen peroxide. So we have large amounts of hydrogen peroxide produced both in the intracellular environment and in the extracellular environment as a result of all of these processes. In honor of Professor Govinji, who has worked so long on photosynthesis, I just wanted to say a quick word about the production of hydrogen peroxide and singlet oxygen by the photosynthetic electron transport chain. In the literature, you will see quite often a statement that the transport of electrons to oxygen is an unavoidable consequence of an electron transport chain operating in an oxygen environment. And for a long time, it was considered that it was uncontrolled production of hydrogen peroxide and singlet oxygen. We now know there are many controls over these processes. And the last 10 years has seen an explosion of information in this regard. So, for example, at photosystem 2, the production of singlet oxygen is controlled by the level of bicarbonate and other carbon compounds which are produced as a result of photorespiration. So even there is control of singlet oxygen production, and all of the electron transport components downstream produce hydrogen peroxide, but in a much more controlled manner than was ever thought possible. So the structures of the complexes which are involved in the generation of hydrogen peroxide and superoxide is regulated in such a manner that the level of hydrogen peroxide can be controlled. So there is a degree of control in all systems of the production of reactive oxygen species within the cellular environment. And similarly, when we come to consider the role of antioxidants and components which process reactive oxygen species, concepts have changed a great deal in the last 10 years. So the classic view is that we have a balance of reactive oxygen species and antioxidants, and that any stress situation perturbs this balance, increasing reactive oxygen and leading to either negative effects or signaling. However, this type of model, while it is effective in describing in simple terms what happens, it is largely incorrect in that it doesn't allow any kind of interpretation in terms of signaling and defense. Whereas if we move to a more modern concept, we actually know now that reactive oxygen species are able to interact with all the proteins in their environment in a way that alters the properties and the oxidation of cysteine groups. And so the signal from reactive oxygen in terms of oxidation state is processed via the oxidation of target proteins which are able to transduce the signal. Similarly, the signal can be interrupted or stopped if the interaction is with an antioxidant. So in any compartment of the cell, the reactive oxygen species that are formed can either transmit a signal in different ways according to which protein interacts with the reactive oxygen species, or whether an antioxidant stops the processing of the signal. And this allows us to consider a model where there are multiple ways that reactive oxygen can be processed to transmit signals in individual cell compartments, to either be through acclimation or cell death. Once this concept is in your mind, then it is quite easy to understand how redox signals, oxidative signals, are processed in individual cell compartments. Because in each cell compartment, for example, like the chloroplast, there are proteins which are there to perceive and process oxidative signals in order to transmit signals to the nucleus to change gene expression in a relatively specific manner. Similarly, we have proteins on the plasma membrane which are able to perceive and transmit signals from the apoplast according to the production of hydrogen peroxide in the apoplast, and to transmit that signal into the cell to change gene expression in the nucleus. One of these proteins, which is now very well characterized, is called HPCA1. This is a hydrogen peroxide receptor on the plasma membrane, and this is a protein kinase receptor, and it can translate an oxidative signal through the activation of kinase activity into a phosphorylation signal within the cell, which then alters gene expression in the nucleus. So we have multiple ways identification of these receptors is relatively new. So it started with HPCA1, but other redox sensors in different cellular compartments are now being identified. And just at the bottom of the slide, there is one here that I would direct you to, which is called QSOX1, which is again a redox sensor within the intracellular environment. But I think in coming years there will be an explosion of the identification of these receptors for hydrogen peroxide in different compartments of plant cells. So this is the background. In measuring redox changes and oxidation, we have classically used biochemical methods to measure hydrogen peroxide and also methods of enzymology and biochemistry to measure antioxidants, whether they're non-enzymic antioxidants or enzymic antioxidants. While these measurements are very useful, they don't tell you anything about. So, for example, if you measure hydrogen peroxide within leaves or other tissues, you get just an overview of hydrogen peroxide across the cell. And as I told you, much of the hydrogen peroxide is actually in the intracellular compartment, in the apoplast cell wall compartment, not in the intercellular compartments such as the cytosol and the chloroplast. So it is more useful, and many labs have turned to using genetic probes, which are continuously improving now. This is one of the classic examples of an in vivo redox sensor, which is a redox sensitive green fluorescent protein. This is a green fluorescent protein which has been modified to have a pair of redox sensitive cysteines, which can be oxidized upon perception of an oxidizing environment. And this changes the peak of emission of the probe. So we were able to calculate the oxidation degree in any given cellular compartment to which this probe is targeted. And this can give us much more precise information about the changes in individual cellular compartments. We have used this probe over many years. As I say, these probes are continually getting better and better. So much so now that it is possible to get probes which are specific for hydrogen peroxide, as well as these probes which measure the oxidation degree within the cell through a measurement of the glutathione redox state. And here you see that in these tissues, these are leaf epidermal cells from Arabidopsis. We have green fluorescent protein. We measure at 2 wavelengths. One is oxidized, one is reduced. But you can see the expression of the green fluorescent protein, in this case, in the cytosol and the nuclei of the epidermal cells. And here we have the stomatal guard cells. Again, you see the expression of the probe in the cytosol and in the nuclei. Once we have this targeting, as I say, the protein can be targeted to any compartment within the cell in order to give you specific information about changes in the redox state within the cell. So we use this information to detect changes in the oxidation of the cytosol and the nucleus in response to various stresses. And it gives me always great pleasure to show this data because it was obtained in collaboration with colleagues from India, and it involved the application of heat stress. So a PhD student from the lab of Professor Anil Grover came to my lab, and her name is Risha Barber, and she was interested in understanding how heat stress changed the redox state of the cytosol within the Arabidopsis seedlings after an exposure of 1 hour to heat stress. So we were able to treat the seedlings and then measure what had happened to the tissues. So here we are looking at our epidermal cells and our stomatal guard cells. And if we just look at the oxidation degree, the white bars are the absence of stress. Excuse me, we'll go back. The black bars are the cells which have been treated with heat stress. And you can see that there is a huge increase in the level of oxidation of the cytosol, but surprisingly, there is a similar oxidation of the nuclei within those tissues. And the same with the stomatal guard cell. There is an oxidation of the cytosol and the nuclei. This type of data was totally unexpected because it had not really been shown that oxidation of the nuclei occurred in response to stress. And we have seen now that oxidation of the nuclei occurs very rapidly in response to many stresses. We don't know exactly the cause of the oxidation of the nuclei because there are very few processes in the nuclei that we know generate hydrogen peroxide, but it does occur. And recently, colleagues from France have presented data, although it's not yet published, showing that oxidation of the nuclei occurs before oxidation of the cytosol in response to heat stress. So this is a really interesting piece of information. And then you can look at the regulation of expression of antioxidants and other systems which accompanies the change in oxidation. There are actually very few changes in the antioxidant system, which is surprising. The only real change comes in one of the forms of ascorbate peroxidase, and you can see this is the ascorbate peroxidase gene family in Arabidopsis. And there is only really a very specific expression of APX2, which is a form in the cytosol and bundled sheath cells upon response to stress. So there is only a very specific response in terms of the antioxidant system to heat stress, as well as the many heat shock factors and heat shock proteins which are generated during this process. And one presumes this is really to allow the progression of the signal to change gene expression. In addition to the effect on the expression of APX2, so we have shown there that there is a change in gene expression. There is, in addition, a change in the activity of the protein, which is generated by interaction with a calcium-specific protein kinase, CPK28, which we showed in this paper here, directly interacts with ascorbate peroxidase 2. We know that APX2 mutants are more sensitive to high temperatures. Similarly, CPK28 mutants are sensitive to more sensitive to heat stress. So if you're interested and look at this paper, we show that CPK28 phosphorylates the APX2 protein, and this increases APX2 activity. So here we have an effect of heat stress on the oxidation of the cytosol and the nuclei, and a very specific effect on the antioxidant system, particularly for APX2 at the level of gene expression and post-translational regulation of activity. In addition to just looking at stresses in leaves, it is possible to look at all of the tissues in the cell, in the plants, excuse me, with these type of probes. And here we're just looking at the tissues of the flower, and it is possible to see the level of oxidation in all of the tissues of the flower, and to quantify this again by oxidation degree. And if we look at the black bars and look at the different tissues of the flower, we can see that they are relatively reduced. The only surprise came with mature pollen. Mature pollen is relatively oxidized in the state before germination. So we have a large state of oxidation in mature pollen. And for the pollen to germinate, it needs to reduce again. So this is germinated pollen, this is the pollen tube. And so there is a change in the redox state of the pollen upon germination. This is just a piece of data to show that the antioxidant systems are important in the flowers, because we are looking here in the white bars at plants that are deficient in an antioxidant called glutathione. And when glutathione is missing, the oxidation degree is increased in all of the tissues, but not enormously, but just to a small extent. And we see this even in the mature pollen. It is more oxidized in the absence of glutathione. And it still is able to reduce upon germination, but this change is sufficient to impede and slow the germination of the pollen. So we can use this type of analysis to really understand more about the redox effects on pollen and other tissues. And again, this type of experiment is now being done with heat stress, because we know that the tissues of the flower are particularly sensitive to the adverse effects of heat stress. So I want to now focus on a piece of work that we have done in my lab related to another process, and this is the sensing of plant growth promoting bacteria by the roots, because I know this is a growing area of interest. Plant growth promoting bacteria and fungi are very important now for agricultural purposes. We have the rhizobacteria, which are important in nodulation and symbiotic nitrogen fixation. We have plant growth promoting bacteria and fungi, and also arbuscular mycorrhiza, which are very important in the assimilation of nutrients by the plant. We know that these types of organisms are really important because they help in the acquisition of nutrients. They act as biological control agents because they can deter negative organisms in the soil. They themselves act as biostimulants because they produce a wide range of secondary metabolites, particularly phytohormones, which are involved in the promotion of growth. And they also are able to alleviate abiotic stress by induction of innate immunity within the plants. So growth promoting bacteria are very, very important. In our own lab, we were interested in looking at the ways that plants perceive plant growth promoting bacteria, with a particular emphasis on how the presence of bacteria changes the redox processes within the root. So these experiments were done with a particular bacterium called Pseudomonas oryzaehabitans, which was identified in the roots of fruit trees and was found to promote growth. We started these experiments simply adding the bacteria to the cultures of the cells, but when you put the bacteria in the same environment or close to the roots, they tend to grow so much because of the exudates for the roots, they crowd the roots. So we went to a different system where we are growing the plantlets on agar plates, and they are separated from the bacteria at a distance. So the bacteria are growing at least 5 centimeters from the base of the roots. We grow the roots we grow the plantlets for 7 days without bacteria, and then we transfer them to plates in the presence of bacteria for a further 7 days and measure what happens. As a result of the presence of bacteria, you can see there are changes in the root system. So root system architecture is changed by the long distance perception of the bacteria, so much so that there is an increase in the number of lateral roots relative to the primary roots, and there is a large increase in lateral root density in response to the presence of the bacterium. And so we asked how the root transcriptome, how the transcript profile of the roots was changed in response to the perception of bacteria. And here are some of the data. In fact, very few changes were observed. You can see here about 400 transcripts were increased in abundance, whereas about 200 were decreased in abundance. And we were very interested in the nature of these genes, because actually we saw very few defense responses. There are some minor responses to stress, etc. here, as you can see, but most of it is response to stimulus, response to chemical stimulus, response to abiotic stress stimulus. And there are large changes which are down to changes in the nutrition of the plants, as well as you can see here a small amount which are relative to hypoxia. And when we look at the specific genes, here we're looking at transcripts which are most increased in abundance, and here transcripts which are most decreased in abundance. You can see the ones which are greatly increased in abundance are largely components which are involved in metabolite processing or the uptake of minerals. So here, for example, we have HAC5, which is a potassium channel which is increased in expression. And strangely enough, the most decreased proteins are these UDP glucose transferases, which the reason for these decreases are not known, but it is a very interesting observation. Just very specifically, we were looking at hormone changes, and actually we didn't see a profile that we were really expecting. We were looking for the hormones that change a root system architecture, such as auxins and gibberellins, and we only saw one auxin transcript. Here, for example, most of the transcripts which are increased in abundance are relative to ethylene, and ethylene signaling. So we have a change in the protein in the transcript profile, which is related to ethylene. We have changes in response to hypoxia, which is perhaps normal because we don't think of this very often. But in a natural environment, organisms are competing for oxygen within the microbiome around the roots. But none of these changes are sufficient to deter or alter the growth of the roots. So we have changes in these transcripts, but they don't affect the actual growth of the roots. They may be involved in root system architecture, and we have to look into that. But here we have this profile of transcripts, which is all related to root nutrition. So you see root hair specific genes here, a sucrose transporter here, various ion channels which are changed. So the long distance perception of bacteria actually manipulates the ability of roots to take up ions and the metabolism of those roots. But that the transcript profile didn't tell us anything about whether there are redox changes, whether there is oxidation. So we went to another system, which is to look at specific mutants and transgenic plants, which are involved in either antioxidant systems or redox processing. And this is one of them. These are plants which are modified in the redox responsive transcription factor 1, RRFT1, which is also called ethylene responsive factor 109. And we have plants which are overexpressing the transcription factor or without the transcription factor. This transcription factor is involved in root architecture, but it also amplifies the ROS signal within cells. So we conducted the measurements as before, and we could see that there was a change in the lateral root density in the control system, but it was lost both in our overexpressors and our mutants. And even in the mutant system, there was some reversal of the nature of the change in lateral root density. So this redox responsive transcription factor 1 is really important in the perception and the response of the root system architected bacteria. We also asked the question, is there a large-scale change in oxidation within the roots, which might be important? And we looked at this in two ways. We took away, in the first instance, the low molecular weight antioxidant ascorbate. So ascorbate is involved in the removal of reactive oxygen species. And here again, you see the control, the increase in lateral root density. But this also occurs in the B2 mutants, which are lack ascorbate, VTC21, VTC24. So we can say that there isn't a general oxidation or a general change in oxidation, which is causing the change in lateral root density. However, there is a specific requirement for the antioxidant glutathione, and we were able to show this in this way. So we have a number of mutants which are deficient in glutathione. You can see here the effect of the perception of bacteria on lateral root density, and it is gone absent when the plants are lacking in glutathione. So we can take this information together now and understand something about the role of reactive oxygen species. We were also interested in looking at the nature of the signal, particularly because we saw the change in ethylene response factors. We wondered if the signal involved in the perception was volatile. So we undertook experiments of this type where we were separating plants and bacteria. For example, here the bacteria are on one side on the gel and the plants are on the other, but they are physically separated. So this experiment would tell us whether the signal is volatile, because if it is a volatile signal, it would still pass, for example, here across the plate. So we did the measurements again in all of those systems, and we could see that the change in lateral root density was still was absent. The volatile signals were not important in the control of root system architecture because the signal was blocked when we separated physically the bacteria from the host plants. So it must be some kind of metabolite signal which is passing physically from the bacterium to the roots. All our data together tells us that what happens upon the perception of bacteria is the importance of redox systems. We know that lateral root density involves redox signals and how they are controlled. We saw the importance of redox responsive transcription factor 1, ERF109, which is required for the amplification of ROS signals and for the integration of hormone signals to control lateral root density. We also saw the role of glutathione, which is very important for components such as these glutaredoxins, which are intimately involved in the control of the formation of lateral roots. What we don't actually know is how the plants perceive the bacterium and how the redox signaling is transmitted. It's considered that within these systems, it's the root cap which perceives signals from the environment. So it's interesting to know now how the root cap perceives signals and transmits the signal through the root to change lateral root density. We also need to rethink our model of how these systems are controlled within the roots, because environmental cues are known to control lateral root density. And the role of our redox responsive transcription factor 1 is very well known. But in the classic view, it goes through hormones such as jasmonic acid, which are involved in altering the expression of ERF109. And we didn't see any transcripts which were involved in jasmonate signaling. So there must be other pathways which are important in this type of regulation. So I would now just like to finish on talking about how cells perceive stress and how stress signals are transmitted through the plants. So as you've heard in all the lectures, plants have to respond very quickly to a wide variety of stresses in the environment. And that perception of stress has to be transmitted and used to trigger processes of acclimation and defense all the way through the plant. So I just want to consider how plants achieve cell-to-cell stress signaling. Just to go back, because it would be interesting to use this information to see how the root cap transmits signals from the environment and whether this happens in the same way. So we are looking at the way that cell-to-cell signaling occurs in plants. And we know that perception of a stress, such as light or any other stress, is perceived by a part of the plant, but signals which change gene expression are able to be transmitted from the site of perception across all of the plant, so that leaves which have never seen the stress and organs such as flowers and roots which have never seen the stress are able to respond quickly by this process of cell-to-cell systemic signaling, which is known to occur to many, many stresses. When for years, for example, it has been known that if insects are involved, or caterpillars which are eating one leaf on a plant, the rest of the plant is quickly responding in terms of expression of genes which deter insect feeding. So this process of long-distance signal transduction requires many types of signal. And we know that hormones and redox processes are involved, electrical signals, calcium waves, and Ross waves, as well as other types of signals such as changes in metabolites, etc. But rather than each of these signals working independently, it now appears that they work in a concerted way to transmit signals. And I will only focus on Ross and calcium waves because these are really, really important. So the transmission of signals from local tissues to systemic tissues involves the generation of reactive oxygen species. And this occurs by a combination of different signalings, but basically what happens, the perception by one cell of a stimulus or a stress activates these enzymes on the plasma membrane called respiratory burst oxidases, and this releases hydrogen peroxide into the apoplastic environment. The hydrogen peroxide signals then moves again into the cell or into adjacent cells to trigger changes in calcium and other signals which open plasmodesmata and allow the movement of other signals such as metabolites and hormones in a systemic way. But the signal, it absolutely requires the production of reactive oxygen in the apoplast. This is called the Ross wave. So this type of process has been shown to be involved in most types of signaling. Every time somebody has looked at a stress type of stress, this type of signal has shown to be important. In my own lab, we've shown that this type of signal is important in response to plant responses to high CO2. So high CO2 responses in shoots are transmitted through the Ross wave to roots to change processes in roots. So the Ross wave is important in acclimation to all types of stress. And this has been shown in many types of response, but the ones that I want to show you now are just very good examples. We know that when plants respond to stress, they often close the stomata. This is a response in light stress, but it could be done also in response to pathogens. So a rapid response to pathogens is the closure of stomata. But here, when one leaf is exposed to highlight stress, there is a rapid closure of stomata in that leaf, but there is also a closure of stomata in systemic leaves. And if you look at the time scale, the closure of stomata in systemic leaves, which have never seen the light stress, is almost as quick as the closure in the local leaf. So there is a rapid movement of the signal from leaf to leaf, which requires the Ross wave. So in the lab of Rob Mittler, they have used imaging of different types of reporter protein to look at how the signal spreads. And they've used this instrument, which is generally used for detection of probes within animal systems. And they have very effectively used this system to measure in real time the progression of signals. And this is a really nice demonstration. So this is the perception of light stress in an Arabidopsis plant through the expression of a reporter. The reporter is green fluorescent protein, which is expressed under the control of a promoter to ZAT12. ZAT12 is a transcription factor, which is expressed in tissues in response to light stress and other stresses. And this picture shows from where the light stress is applied with the arrow, there is rapid movement of the signal for the expression of this transcription factor through all the other leaves, through the flowers, and across the plant. So we can see really the passage within real time of the stress signal from the point of perception across the plant. And we know the components that are involved in this signaling system because of work in many labs, but here is some of the data from Professor Fleischmann, which I think is very nice. This data shows plant responses to different types of stress in terms of the expression of ZAP12. And you see here highlight stress, the timing here, 0, 5, 10, 15 minutes. You see the expression of the transcription factor from the point where the stress was applied across all of the plant. Here we have a different type of stress. We have wounding of this one leaf, which causes a systemic response very, very quickly. And here we have the application of a pathogen. And so each of these stresses is causing a systematic response that requires the Ross wave. We know that the Ross wave is required because we can delete components of the system and we can alter the signal. So here is the RBOHs in the plasma membrane, which are required for the production of hydrogen peroxide in the apoplast. And here we're looking at, again, light stress in the wild type, but in mutants which lack one form of this enzyme, RBHD, and you see the signal is lost. The systemic signal is much weaker. Conversely, if we take a mutant lacking part of the antioxidant system, this is APX1. Now the antioxidant system will of course impair the signal, so the presence of antioxidants is there to police the signal, and you can see that in the wild type on the top line we have the response to the light stress, but it is much amplified in the signal where we have removed part of the antioxidant system. So in fact, the signal is transmitted much faster in the absence of antioxidants. You can also measure this type of systemic signaling using the ROGFP that I showed you earlier. So here we have the oxidized, the reduced form, the stress is given here. And you see here a time-lapse photo of the increase in spread of oxidation after one leaf is targeted with a light stress. So even with this probe, you can see the systemic spread of oxidation across the plant to spread the signal. So using this type of experimentation, we can see that there is a really important wave of signals which moves from cell to cell that involves the apoplastic production of hydrogen peroxide, which is completely dependent on the presence of respiratory burst oxidases. And using different types of mutants across the in different labs, it's now been shown, for example, in Ron Mittler's lab, that HPCA1 is essential for the perception of the hydrogen peroxide signals, that aquaporins are required, the opening of aquaporins is required for transmission of redox signals into cells, and that the opening of plasmodesmata via this process is also involved in the movement of other molecules from cell to cell to spread the signal. Of course, there are many, many components that are involved in this process. And quite recently, in fact, it appears that components that we never suspected, like phytochrome B, are really important in the transmission of signal, even in the transmission of light signals, and that actually production of ROS5 chloroplasts isn't so important in the transmission of light signals. Rather, it is the production of RBOH in the apiplast, the production of hydrogen peroxide by the RBOH in the apiplast, which is required for the transmission of light signals even through the plant. So, more or less, I would just like to conclude there with a summary. I hope that I've convinced you that reactive oxygen species play a wide range of roles in plant cell, but they are particularly important in plant responses to stress. They are important both in the local responses and in the systemic responses, which are required for systemic acclimation. And this involved changes not only in defense, but in the development and growth of the plants, as I showed you in the root system architecture. We know some of the components which are involved in systemic signaling, but there are many components which are still to be discovered, many components which we still need to find are involved, because obviously this is a complex network. I've only told you about basic cell-to-cell signaling. What I've told you tells you nothing about the specificity and the nature of signaling. And of course, we have many components to be identified, which will be involved in the generation of specific signals. So in conclusion, I would just like to thank all my collaborators. Here are some of the PhD students in my lab who I thought you might like to see. My collaborators, particularly Ron Mittler, and colleagues in Spain and in India. Thank you very much for your time, and thank you for listening.
Thank you so much, Madam. It was, in fact, a great talk on the roles of reactive oxygen species in plant growth, defense, as well as development. Previously, the reactive oxygen species were considered as something evil to the plant system. And as of now, your studies, and they have very clearly shown how these systems are optimizing the development process in plants in different organelles. And thank you so much. You have given a very broad outlook on how the things are working, and introduced the newer term, Rose wave, and also presented the current model for the Rose wave. Thank you so much. And
thank you.
To the house, any questions? Yeah, please. Yeah, just one question.
Christine, a very nice talk. I think we learned a lot. As usual, your talk is always a treat for us. So I was just wondering, when you talk about this cell-to-cell signaling, and you talk about RBOH gene playing a very important role in this communication of ROS between cell to cell. Now, ROS is actually having multiple type of, RBOH is of multiple, like RBOH-B, RBOH-I, RBOH, and all of that. And these are very, very cell-specific, I would say, tissue-specific. So how do you see the communication in light of being specificity within the RBOH system, and what is the current understanding for this?
Yeah, as far as we know, RBOHD and RBOHF are really the main components which are involved in cell-to-cell signaling. The other RBOHs are very much involved in specific processes, but in fact, they could be involved in the transmission of signals in very specific cell types. Most of the work that we have shown you has actually just been in leaves or in roots. So, because it's easiest, I guess. Right. Some of the more discrete signaling, for example, that would occur in flowers or in other in other tissues, might rely on other RBOHs, but that just has not been investigated.
Right. I think it's very intriguing to see that how the minor structural differences are bringing in the specificity with respect to the vegetative state or tissue specificity or reproductive. I think this field is opening up in a big way now, I would say.
Yeah, there is so much to do, Ashwani.
Yeah.
You know, I've shown you just very basics. We've really just scratched the surface.
Sure.
The more we learn about the components, the more we will learn about this tissue specificity and the stress type specificity.
Sure.
Because there will be modulation of this signal by different stresses.
Well, thank you very much for the nice lecture. We enjoyed it.
Thank you, Ashwani.
Christine, Jos Putur here. So,
Hi, Jos.
Yeah. So, thanks a lot for joining with us. Of course, 5:30 was too early for you. Now, I think now it is convenient for you. Okay. So now a Momento is waiting for you, but unfortunately, we cannot deliver at this point. Certainly, it will be here, and we will get you here physically once, and it will be given to you. Okay. Next time your visit to India, you are going to come to Calicut. We will plan it that way in your next visit. Hopefully in February, when he comes, you come for the conference of Ashwini, maybe we will plan it out. Okay. So thanks a lot for joining, and now we will move to the validatory session, the concluding. So you were the concluding session. Okay, it's fine.
Thank you so much. Goodbye.
Thank you so much, and we are concluding the session.
Now, I would like to welcome Dr. P. Sunoj Kumar, Associate Professor, Department of Botany, University of Calicut, to present the memento as a token of appreciation to Dr. Deepu Matthew, Chair of the session. Sir, please.
This is a small announcement. We will not break for tea. Hopefully, we are trying to serve the tea here. Because due to limitation of time and our Chief Guest, Pro Vice Chancellor, has to leave at 12:30, we will not break for tea. So be in the hall itself. Thank you.
Dr. Sumesh KV, co-chair of the session. Dr. Fasila P, session rapporteur. Thank you, sir. Thank you, ma'am. Ladies and gentlemen, we are pleased to welcome you to the valedictory ceremony of the International Conference on Physiological and Molecular Mechanisms for Abiotic Stress Tolerance in Plants, jointly organized by the Indian Society for Plant Physiology, South Zone, and the Department of Botany, University of Calicut.
Now we are soon going to start with the valedictory session, and after tea, Dr. Ashwini can be on the stage. You can have it slowly after that. Yeah, then a program.
The chief guest of the day, Honorable Pro Vice Chancellor Dr. M. Nasser, University of Calicut. Distinguished Professor A.S. Raghavendra, Professor, University of Hyderabad. Respected Professor Ashwini Parikh, Executive Director, National Agri-Food Biotechnology Institute, Mohali, Punjab. Distinguished Professor Om Prakash Dhankar, University of Massachusetts, Amherst, USA. Beloved Dr. V.V. Radhakrishnan, Professor, Department of Botany, University of Calicut. Convener of the program, Professor Jose T. Puttur, Head, Department of Botany, University of Calicut. Dignitaries, on of the dais, eminent scientists, professors of various institutes, teaching faculties and non-teaching staff, research scholars and students. Once again, welcome to the valedictory ceremony of the International Conference on Physiological and Molecular Mechanisms for Abiotic Stress Tolerance in Plants, jointly organized by the Indian Society for Plant Physiology South Zone and Department of Botany, University of Calicut.
Let us start the ceremony with a prayer. I invite our PhD student, Ms. Aparna Srikumar, for the prayer.
വിശ്വേകപാലക വിജ്ഞാനദായക വിഘ്നങ്ങളെല്ലാം അകറ്റിയിടണേ. മാനസ പുഷ്പങ്ങളർപ്പിച്ചു നിൻ പാദപത്മങ്ങൾ ഞങ്ങൾ വണങ്ങിടുന്നു. മാനസ പുഷ്പങ്ങളർപ്പിച്ചു നിൻ പാദപത്മങ്ങൾ ഞങ്ങൾ വണങ്ങിടുന്നു. വർണ്ണങ്ങൾ മെയ്യുമി സ്വർഗീയ ഭൂമിയിൽ വായുവും വെള്ളവും നീയല്ലയോ. വിണ്ണിലെ സൂര്യനും ചന്ദ്രനും പിന്നയ താരാഗണങ്ങളും നീയല്ലയോ. ആ മഹാശക്തിയെ കൈതൊഴുന്നേ, ആ ദിവ്യ തേജസ്സെ കൈതൊഴുന്നേ. ആ മഹാശക്തിയെ കൈതൊഴുന്നേ, ആ ദിവ്യ തേജസ്സെ കൈതൊഴുന്നേ. ആ ദിവ്യ തേജസ്സെ കൈതൊഴുന്നേ, ആ ദിവ്യ തേജസ്സെ കൈതൊഴുന്നേ.
Thank you, Aparna. To welcome the gathering, I wholeheartedly invite beloved Professor Dr. V.V. Radhakrishnan, Dean Faculty of Science, former Head, Department of Botany, University of Calicut. Welcome, sir.
Good morning, ladies and gentlemen. My duty is to welcome you all to this valedictory function. We have come to the fag end of this 3-day international conference. And as you are aware, climate change is a fact. And climate change has affected our crops and plants is also a fact. So now we have to think and plan means to overcome this situation. Last century, it was food scarcity, and our agriculture scientists across the world, they have developed varieties, developed packages, developed measures for overcoming our issue of food security. Now the problem is we are not in a position to sustain the food security because of change in climate. So the duty assigned to scientists and researchers is to work out ways and means to overcome this grave issue by equipping our crops and plants to adapt with the changing climate. So last 3 days, our eminent scientists and researchers across the world were assembled here. It has been a conclave of eminent plant physiologists within the country and abroad. And I'm sure that the participants and delegates got a lot of ideas about how to solve this issue. And what I learned from many of the participants, that they could get a lot of information, they could get a lot of ideas, a lot of innovations, so that they can sharpen their experiments and studies to suit the changing pattern of climate. So my duty is to welcome the guests as well as the participants. So today, valedictory address will be given by Professor M. Nasser sir. He is a good friend of our Department of Botany, basically an entomologist. So being an entomologist, he keeps a love towards plants, and he has been supporting us right from 1984, more than nearly 4 decades. So all the success of our department is because of his support for all academic as well as research activities. So on behalf of the Department of Botany and all the delegates assembled here, and on behalf of the organizing committee, I extend a warm welcome to Nasser sir. So I was told by my friend and colleague, our Joe sir, that we have only very limited time for the valedictory function. So I don't want to speak more. So I invite and I extend a warm welcome to all the dignitaries and experts and tall scientists on the dais and off the dais to this valedictory program. And the program went on well, and the experience, the participants, they gathered during the last 3 days, I think these memories will remain green in their heart even after they go out from the campus. So I wish you all a happy return journey and best of luck. Thank you.
Thank you, sir. It gives me immense pleasure to welcome esteemed Chief Guest of the Day, Professor M. Nasser, Honorable Pro Vice Chancellor, University of Calicut, to deliver the valedictory address. Sir, please.
Respected Head of the Department, Dr. Jos Puthur, Sir Dr. V.B. Radhakrishnan, Dr. Muhammad Nasser, Professor Raghavendra, Professor Ashwini Parikh, Professor Om Prakash, Dr. Shakira, other dignitaries on the dais and off the dais, delegates to the conference, faculty members of the Department of Botany, dear participants and dear students. As an entomologist, I'm always in love with plants because, as you know, insects and plants have always been interacting for all these years. And there is no winner, I think. It's always interaction. So I always love to come to the Department of Botany for any of the function because I need to keep a close watch on what plant scientists are doing. Because from an insect point of view, it's better I get to know the plants. So it's indeed a pleasure to be here for addressing this August gathering of eminent scientists and research scholars in plant science. So I consider it a privilege to deliver the valedictory address on this timely and highly relevant topic, physiological and molecular mechanisms, markers for abiotic stress tolerance in plants. So I am sorry I could not follow the discussion during the past 3 days. But as we all know, climate change is impacting every living organism on this earth. And as an entomologist, the study on the impact of climate change on insects is, I suppose, still in the nascent stage. We have not specifically determined what the impact of climate change is on insects. We know its diversity is affected. We know lots of effects are there, but the exact mechanism of this impact is not known. But I hope plant scientists are far ahead on this, and they do know how the impact of climate change on plants is. Because it's very important, because plants provide us lots and lots of goods and services. So getting to know the direct impact of climate change on plant, on the abiotic stress, will influence how our growing population will be fed with whatever food we want. Because food is a problem for our growing population. And the provision of food comes from plants. And if plants are affected by climate change, that affects our population. So I'm sure during the last few days, you had real good discussions and real good outcomes are there so that we can mitigate the impact of climate change on our plants. So I also commend the Department of Botany and the Indian Society for Plant Physiology for organizing this exceptionally relevant seminar. So I'm also glad to know that we had the participation of foreign delegates and lots of participants from outside Kerala came over here. I hope you had a very good stay over here. You enjoyed it, because usually botany departments are good organizers and good hosts, or they may have taken good care of you. So to the budding scientist over here, I hope you had the opportunity to present your findings. You had the opportunity to interact with the eminent scientist over here. And whatever knowledge you have gained from over here, I hope you will use it for the betterment of our society. Because we need to take care of each and every living organism on this earth. So whatever ideas you have got from here, whatever knowledge you have gained from here, I hope you will put into practice, and your further future research will focus on that for the betterment of all the living organisms on the earth, because we know we have only one earth and we should take good care of it. So I also wish you all the best in your future endeavors, and also once again congratulate the winners of the poster presentation, the paper presentation, and also once again commend the Department of Botany for organizing such a seminar in such a systematic and beautiful way. Thank you very much.
Thank you, sir, for your words of encouragement. Next, the Department of Botany is fortunate enough to have an international participant for the conference, and we would like to express our appreciation and gratitude to Dr. Muhammad Nasser Al-Yamani, Professor, King Saud University, Riyadh. I invite Honorable Pro Vice Chancellor, Professor M. Nasser, to present the memento to Dr. Muhammad Nasser. Sir, please.
Just a word about him. He is about to retire, but still he was very keen to attend this conference, and I remember since 2 months he is contacting me regularly, and he had a lot of hurdles, because when he applied for the visa, he didn't get the visa because of some obstruction from Ministry of Home Affairs and External Affairs. But he kept on trying, and because seeing his keen interest, I took special interest and applied for that, and finally all certificates we received, and finally he got the visa 2 days back of the conference. So he is especially honored for that, for taking the great interest in attending our conference. Thank you.
Thank you, sir. Next is the most anticipated event, the announcement of winners of the various oral and poster presentation competitions and award distribution. I would like to invite Professor Josri Puttur, Head Department of Botany, University of Calicut, to announce the winners. Welcome, sir.
Yeah, this session is put a little in front just because our Vice Chancellor has to leave very soon. So we'll complete this task, then we'll go into the next sessions. So, and another word regarding the competitions and presentations. We had 4 themes altogether in this conference, and impact on photosynthesis, power of phenomics, strength of redox system, water relation, and mineral nutrition. That was the 4 main themes which has been put up for this conference. And we had, altogether we had around 49 presentations in the poster as well as oral side. But certain categories didn't have enough of competence, or there were no minimum participants, and because of that we could not give away the prize in that. But almost 12 prizes are being distributed over here. So you may feel that why there is 12, since 4 into 2 it should be 4 into 4 it should be high, because in each category we have the theme, we have the senior category as well as the junior category. So taking it that way, it would be around 16, but still only 12 are being distributed. That is because we have some minimal competitions in some, and because of that we are not distributing. Yeah, and the results of the theme impact on photosynthesis, the best oral presentation in junior category is Muhammad Yusuf Zamal from University of Hyderabad, Department of Plant Sciences. And I invite Professor Nasser to give away the prize to Muhammad Yusuf Zamal. He's here? Okay, maybe he must have left. The second one goes to, for the poster presentation in the senior category, Muhammad Sadiq from Rubber Research Institute of India. Yeah, our Professor Nasser, please. Again, in the theme of impact on photosynthesis, best poster presentation in junior category, Jerome Xavier from University of Hyderabad. Jerome is here. Or somebody from Hyderabad, University of Hyderabad can have the, you all have left? That is very bad. In the second theme, power of phenomics, best oral presentation in junior category, Sharat G. Nair of Department of Botany, University of Calicut. The best poster presentation in senior category, Krishna Priya from ICR Sugarcane Breeding Institute, Coimbatore. Now in the best poster presentation in junior category, Navneeta S. from Kannur University. Now may I invite to distribute the next 2 awards by Professor Raghavendra to best in the theme of strength of redox system, Best oral presentation in senior category, Dr. C. Apunnu of ICR Sugarcane Breeding Institute, Coimbatore. Best oral presentation in junior category, Shashibhushan Gahir from University of Hyderabad. Then best poster, now I may invite Professor Ashwini to give you the prizes for the best poster presentation in junior category, Sonal Sangwan from Central University of Tamil Nadu. Now the next theme, water relation and mineral nutrition, best oral presentation in senior category, Dr. Bhuminathan from Tamil Nadu Agricultural University. May I invite Professor Om to distribute away the next two awards. Best oral presentation in junior category, Jayendra Pandey from University of Hyderabad. University of Hyderabad has bagged the maximum, but not a single person is present here. Yeah. Maybe the emeritus professor can give them all these prizes together. Best poster presentation in senior category, name, yeah, Dr. Fasila P, Unity Women's College, Manjeri. She is alumni of our Department of Botany, University of Calicut. Now she is assistant professor in Unity Women's College, Manjeri. Now for the next and the last award to be distributed, may I invite Professor V.V. Radhakrishnan, the Dean Faculty of Sciences of our department, our university. Best poster presentation in junior category, Nigil PT, BITS Pilani, Hyderabad campus. As our Pro Vice Chancellor may leave soon, I just want to give away the special recognitions of the arrangements what has been made for this conference. You must have seen the very beautiful arrangements done, especially with regarding to the thematic area, which is commonly the students were telling the selfie spot. And all the arrangements has been arranged by Frames Group, and I invite Assis, if he is present here, to receive a memento from the Pro Vice Chancellor for his arrangements made for this conference. The whole video streaming, the video wall, all this beautiful arrangement has been done by him. So a special recognition is given to him. And also the good food served by Fiesta Group. They are also being recognized here because everyone has given very good comments about them, the food they have served. So Iqbal of Fiesta Catering is also invited to the stage.
Thank you, sir. And winners, congratulations. Moving on, I would like to invite respected Professor A.S. Raghavendra, University of Hyderabad, to share his experience and valuable suggestions on the conference. Sir, please.
A one-word statement about the conference: overwhelming. I think it was done beautifully and efficiently in several ways. As already you have seen the recognitions, everything was perfect in the sense arrangements, transport, food, accommodation and programming and coordination. I think fantastic. That's on the what you call as overall individual side. But any conference is meant for distribution of knowledge, triggering cooperation, and also igniting young minds. I think all these aspects were quite well taken care of. I could see the interaction and the enthusiasm among the youngsters who were here. And then a special word of appreciation to Professor Jose for having taken the lead and done such an efficient and wonderful job. And your also special recognition and special mention should be made of all the students who have done. You know, it was flattering when they come sir, please have your seat. That was moving, I will say. So, special thanks to all of you, students who are taking care of all of us. So, and of course, what we can do is try to mimic and learn from the kind of organization you had. And I think hats off to you, you have done well. We, at the University of Hyderabad, we will also try to implement and learn quite a few things. Our students are also able to do it, and that's really nice. Thank you so much again, once more.
Thank you so much, sir, for your words of appreciation. Next, I cordially invite distinguished Professor Ashwini Parikh, Executive Director, National Agri-Food Biotechnology Institute, Mohali, Punjab, to speak a few words and share his experiences during the conference. Welcome, sir.
So, very good afternoon to all of you. Respected Pro-Vice Chancellor and other dignitaries sitting on the dais. I think for me, I would like to put on record my sincere thanks to Dr. Jose Puthur and everyone who's behind the show. I know Jose Puthur is the main person who's on paper, but when it comes to handling the things behind the paper, behind the curtain, I know many of you have had sleepless nights and a lot of planning it requires. I realize it because we are planning to organize a similar conference in January, so I can very well understand how much commitment, how much dedication and how many effort does it require to organize a successful event. So I will not repeat what Professor Raghavendra has said, but I would like to draw your attention to one of the things before I go back. This conference is actually very, very relevant in today's context, because if I give you one example, we could not conduct census in the year 2021, but based on the census 2001 and 2011, if I give you one figure, that should be very alarming. If you look at the census of 2001 and 2011, you see a lot of parameters. When you do census, a lot of parameters government gave. One of the parameters which I was reading through was very, very alarming. In India, we are losing 4,432 farmers every day in our country, meaning that by that 4,432 farmers are discontinuing farming in our country every day, and this has been happening for the last 15 years. I do not know what is going to be there in 2021, because we could not conduct the census in our country because of Corona. But this gives you an idea that how much important the farming is and what should be done, not only in terms of science, but in terms of policy matters as well. We need to look after the farmer. It has to be a lucrative business. Return of investment has to be there. And one of the way we can do that, to understand the physiology, the biochemistry of plant, and give them suitable seeds so that they are able to continue farming even under these changing climatic conditions, and when we have to feed 9 billion people in years to come. So I think I would like to put on my sincere thanks to Jose Puthur and the whole team for organizing such a wonderful event. They have been talking to me every now and then. They have been sending emails and pressurizing, and I'm very happy that I could make it and meet a lot of people who have really contributed to this science. Thank you very much once again, and I wish you all the best.
Just a minute, we want to give recognition to our respected judges who judged the poster and oral presentations. The paper just came in my hand right now. Actually, we are only giving the mementos to those who didn't receive mementos as chair or co-chair. Some have already received. We had more judges in all the sessions, but I think it will be some 12 into 2, that will be around 24 judges, but only 6 are being given here. So one is Dels P. Sebastian is here from Devagiri College. He is the faculty there. He's not here. Dr. Binu from Forestry, College of Forestry, Kerala Agriculture University. Yeah. Our Pro Vice Chancellor will be distributing. And Dr. P. Sharmila is another judge we had for the session. Another judge is Minu Divagaran from Providence College. I think she's not here. Dr. Yogesh Mishra from BHU. And Mohammad Sadiq, doctor, has to again come up onto the stage to receive the memento. From Rubber Research Institute of India, Kottayam. Okay, thank you, sir.
Thank you, sir. I wholeheartedly welcome respected Professor Om Prakash Dhankar, University of Massachusetts, Amherst, USA, to grace us with his words. So please.
Good afternoon, everybody. Wow, what a fantastic 3 days of science marathon we had here. And lots of great talks and lots of good posters. And also, mainly for the youngsters, what a great opportunity for networking. Apart from good science, the other purpose of the conferences like this is networking, so that you can establish collaborations and then that's how the science grows, by collaborations. So I'm sure many of the youngsters have made good connections with everybody, and they will follow up those connections and implement those ideas that you have and the knowledge you have gained from here into your own projects. The other aspect was, actually the beautiful part of that one is, we were all in person after a long time. And the online conferences, yes, we can hear the talks, but the networking part was missing. Seeing each other, talking to each other, that was missing. So that was the great opportunity we had here. And also, I would like to congratulate Dr. Jose Puthos and his team, entire botany department. A fantastic arrangement. And I know it's not easy. It's a lot of efforts have gone into this one, day and nights. And particularly the students, how wonderful they were, and they are here. They were so respectful, so friendly. They help everybody, whatever they help, any audience or delegates need, they provided that. That's a hats off to you, all of you. Thank you very much. And also, I would like to again convey my sincere thanks to Dr. Jose Puthur and Dr. Shakira to invite me. And my experience was not only the great experience in the conference I had. They arranged my lectures at 3 different places. My first interaction, a half-day interaction, was at University of the Kannur University, and that was the good experience. You can see I was amazed by the 90% participants. I said yesterday 90% participants were female, so it was really good. Kind of see the women, girls are more in biology. That's good, a good thing. And then yesterday I went to Providence College, and that's the only woman, that's the woman-only college, and I think it's the first in Kerala, right? And that was a fantastic experience as well there. And I think other than that, just wish you all the good luck in your science endeavor and your scientific journey, and hopefully we can see each other maybe at in January. Many of you may be coming to January meeting in Nabi. We will see you there, and have a safe journey. Thank you.
Thank you, sir. It is of great privilege and honor for the Department of Botany, University of Calicut, to host the International Physiological Conference of the Indian Society for Plant Physiology for the first time in Kerala. We thank all the participants for making it a grand success. We would like to have the valuable feedbacks and suggestions from the participants, and that is what keeps us going forward. I request the participants and delegates of the dais for their comments. Participants, please.
1 minute in between. I think University of Hyderabad is back. Yeah. So, yeah, just a minute. You can represent the... Yeah, I think all of you can come over here, and University of Hyderabad prizes can be distributed all together. Please come. Yeah, please come to the stage. University of Hyderabad. Yeah, the first one is Muhammad Yusuf Zamal for the best oral presentation in junior category and impact on photosynthesis. Then Jerome Xavier is here. His best poster presentation in junior category, University of Hyderabad. Shashibhushan Gahir, University of Hyderabad for best oral presentation in junior category in strength of redox systems, under the theme strength of redox systems. Jayendra Pandey is here. He is in the category of water relation and mineral nutrition, best oral presentation in junior category. Thank you.
Thank you, sir, and congratulations, winners. Now I would like to request the participants and delegates of the dais for their comments, feedbacks, and suggestions regarding the program. Participants, please.
Hello. Please come up.
A very good afternoon to all of you. I can share my experience in a very few words. Since I have been participated in many national and international conferences, and I would say this conference is standard of any international conference where people from all the age group, eminent in their particular area, and for me, a person of basic stress biology, it was a very good experience to learn that how many people are working with the crop plants, orphan species. Maybe that I am going with the idea that, okay, there are few people in Tamil Nadu Central University who is doing great. So as a young assistant professor, I can find some opportunity to interact. Similarly, because I travel long way, like 4-hour journey in flight and in between 7-8 hours, so it was an overwhelming experience, and above all, I would congratulate the Department of Botany, because many times people say that we are a family, but here they executed, they are the family. every person, every student was showing commitment that we have to do our best. That is the learning which I want to translate in my department whenever any incidence is going to happen. So we have to work like that. And I find that every volunteers was feeling, sir, may I help you? Do you have any problem? Even yesterday night, 2 people, I put a WhatsApp message that I'm not finding bus on the gate. 3 people immediately come and 1 person called. So this is the showing the commitment of the volunteers. So congratulate all the guys who was behind the curtain and for the big success of this seminar. And thank you, Professor Jose Puthur, sir, for giving this opportunity. And I'm speaking non-diplomatically and I'm going with the very good memories. Thank you very much.
Krishnapriya ma'am was not here and now she is present. So we want to re-announce the best poster presentation in the senior category. Krishnapriya, Aika, Sugarcane Breeding Institute, Coimbatore, to receive her award from the dignitaries.
I want to just say one more thing, just quickly, to the youngsters, basically. You are in a very good field. Sometimes people think that IT and all these are getting good jobs. I can tell you now, from my own experience and where the next innovation is going. Next innovation, next 2 decades, the innovation will be the agriculture innovation. And IT innovation and all these, we have done that in the past 1 or 2 decades. The next innovation will be in agriculture. So you will have a lot of opportunity, jobs in the private sector, startups and all these. So you are in a very good field. Just keep it up and stay inspired. Thank you.
Thank you, sir. We are continuing with the feedback session. I encourage the participants to share your valuable feedbacks and suggestions.
3 are compulsory, 1 is over. 3 are compulsory. Yes, you made it 3 compulsory. I thought the same time. Yes, 1 more get prepared.
Very good afternoon to all. As my previous colleagues said, the meeting had been a very overwhelming, as Dr. Yeah, it's okay. It was very overwhelming because I should say that I was wondering that because it was near Deepavali, so I was wondering whether people from North India or other states will come or not, because except Kerala, all other states are celebrating Deepavali. Kerala, it is lesser only. So I was wondering people will come or not, but after coming here, I saw that it is really, you have made it. Actually, the amount of care they have given to receive the people from the airport or railway station or bus stand and put them in the respective places, hotels or guest houses, they have taken such a care like anything, because I can see more than 30 people, 30 students are here. As previously said, they were taking care of each and every one. Like they were, please come sir, please sit sir, or take your seat sir, please take food, everything. They were attending to everything. All of our needs were addressed. And I was really, as said, overwhelmed. It is really a good experience. More than that, I felt that the campus, I have never seen such a nice campus in Kerala other than this. Probably one of the best campuses in India.
and one of the best in Kerala, I would say, because we can see in even the botanical garden or the park, everywhere, very much care has been given in every angle, every aspect of in maintaining all those things. Special thanks to Dr. Santosh and Dr. Pradeep, who are taking care of this. And the special thanks goes to Dr. Jose Puthur, the one and only Jose Puthur, because he has taken such a care in each and every angle of all the aspects of conducting this seminar in a very successful manner. The persons he has invited, the eminent persons in science, in biology, biotechnology, plant sciences, everybody who have come from abroad and as well as in India, it is really overwhelming and very fantastic experience. As previously said, it is a homely experience. I really thank all the students especially and the faculties and the whole management of the university. Thank you so much for giving a very nice and memorable experience. Thank you.
There is a special request from the delegates. Since the University of Hyderabad has bagged most of the prizes, at least one among the participants from University of Hyderabad is requested to give a feedback.
Good afternoon and all. It has been a very nice international conference I have attended till now, whatever I have attended, and I am very thankful to the organizing committee and particularly the volunteers who have been here. They helped quite a lot, and they were like they were able to see, take care of each and everything what we were requiring here. So thank you one and all, and thank you the organizing committee, and also to the delegates. Thank you one and all.
Thank you all for your valuable suggestions and wonderful feedback. We really appreciate it. And thank you all once again. Next, I would like to invite Dr. Shakira AM to present the report of the 3-day international conference on physiological and molecular mechanisms for abiotic stress tolerance in plants. Ma'am, please.
Thank you. Respected dignitaries on the dais, Professor Jose sir, Om sir, Ashwini Parikh sir, Nasir sir, Raghavendra sir, V.V. Radhakrishnan sir, and all the dignitaries of the dais. Let me very quickly brief the report of this 3-day international conference. The International Conference cum workshop on physiological and molecular mechanism or markers for abiotic stress tolerance in plants was opened grandly with flag hoisting on 26th October 2022. The conference was hosted by the Department of Botany, University of Calicut, in association with Indian Society for Plant Physiology, ISPB, and was financially assisted by Kerala State Higher Education Council, KCSTE, DST SERB, INSA, and BRNS. All the delegates, gathered from 6 different countries and prestigious universities, were formally welcomed by Professor Jose Puthur, the General Convener of the conference and HOD of Botany, University of Calicut. Professor M.B. Chetty, ISPP President and former Vice Chancellor of UAS Dharwad, Karnataka, presided over the session. The International Conference cum workshop on physiological and molecular mechanism or markers for abiotic stress tolerance in plants was inaugurated by Professor M.K. Jayaraj, Honorable Vice Chancellor of University of Calicut, by lighting the lamp. Former professors of plant physiology and meritorious professors of Department of Botany, University of Calicut, were honored during the inaugural ceremony. The abstract book of conference papers was released by Professor M.B. Chetty, President of ISPP. Dr. R. Gomadi, South Zonal Secretary of ISPP, briefed the present status of ISPP. Professor P. Parthasarathy, Department of Environmental Science, Andhra University, felicitated the function. Professor Santosh Thambi, Department of Botany, University of Calicut, proposed the vote of thanks. After the inaugural ceremony, the first technical session of the conference was enriched with 2 invited lectures. A lead lecture by Professor Sergei Shabala, School of Agricultural Sciences, University of Tasmania, Australia, and a special invited lecture by Professor Om Prakash Dhangar, University of Massachusetts, Amherst, USA. The session was moderated by Professor Joni Thopil and chaired by Dr. R. Gomadi and Dr. Abdusalam A.K. The session rapporteur was Dr. Shakira. After lunch, the technical session 2 started with second lead lecture by Dr. Umesh Bageshwar, Texas A&M University, Texas, USA, and a special invited lecture by Dr. Maddi Vanaja, Principal Scientist, ICAR Central Research Institute for Dryland Agriculture, Hyderabad. The session was moderated by Professor V.V. Radhakrishnan and chaired by Dr. Roy Stephen and Dr. Ashish K. Chaturvedi. Dr. Swapna K.S. was the session rapporteur. After the 2 technical sessions of invited lectures, poster presentation of the participants started, and there were about 41 posters from various thematic sessions which was displayed and evaluated in the botanical garden. The Legend of Photosynthesis, Professor Govindji, Department of Biology, Department of Biochemistry and Center of Biophysics, University of Illinois, Urbana, USA, was honored on the occasion of his 90th birthday celebration. Professor Govindji was online and also delivered his keynote lecture. The first day of conference was winded up with the cultural programs by Kerala Kalamandalam team and the performance by our own students. On second day, 27th October 2022, the international conference began with the 3rd technical session with 2 invited lectures. First one, a lead lecture by Dr. Sylvia Toth, Senior Scientist, Biological Research Center, Hungarian Academy of Sciences, Institute of Plant Biology, Hungary. And a special invited lecture by Dr. Dinagar Challabatula, School of Life Sciences, Central University of Tamil Nadu, India. The session was moderated by Dr. Manju C. Nair and chaired by Dr. Anil Kumar and Dr. Pavati Srikumar, and Dr. Dhanya Thomas Titi was the session rapporteur. Technical session 4 was started with the lead lecture of Professor A.S. Raghavendra, School of Life Sciences, University of Hyderabad, and by Professor Rajagopal Subramaniam, School of Life Science, University of Hyderabad. The session was handled by Professor C.C. Harilal and chaired by Dr. Hebbar K.B. and Dr. Krishna Kumar G. Dr. Shruti P. was the rapporteur of the session. It was followed by 3 short lectures by Dr. Shailendra Pratap Singh from Banaras Hindu University, Dr. Prabhat Kumar Sharma from Goa University, and Dr. Rajesh Mehrotra from Birla Institute of Technology and Sciences. After the lunch, the technical session 5 started with Professor P.S. Krishnan Memorial Lecture by Dr. Babu Valliodan, Assistant Professor of Molecular Biology and Genomics, Lincoln University, USA. The session also had 6 short lectures by Dr. Arun Kumar R. from ICAR SBI, Dr. Lakshman R.H. from ICAR IIHR, Dr. Yogesh Mishra from Banaras Hindu University, Dr. Ratnakumar Pasala from ICAR IIOR, Dr. Sharavanan Raju from ICAR CRI, and Dr. Vaibhav Lahot from IINRG. The section was handled by Professor A. Yusuf and was chaired by Dr. Abida P.S. and Dr. Anu Agustin. Dr. Sinisha A.K. was the session rapporteur. Oral presentation of the participants were held after that parallelly at 5 seminar halls, and there was about 44 participants from various universities. On third day, 28th October 2022, the session began with the technical session 6. There was 2 lead lectures, first one by Professor Parthasarathy, Department of Environmental Science, Andhra University, and second one by Professor Ashwini Parikh from Parikh Sir, Executive Director, NABI. The technical sessions were concluded by Professor Christine Helen Foyer, School of Biosciences, University of Birmingham, UK, by delivering the concluding lecture. Dr. Manju C. Nair handled the session and was chaired by Dr. Deepu Matthew and Dr. Sumesh KV. Dr. Fasila P. was the session rapporteur. So with this, we have come to an end of this 3-day international conference. And on my personal behalf, let me thank all of you for your cooperation to make this event a grand success. Thank you all.
I would like to invite the convener, Professor Jose T. Puthur, Head, Department of the Botany, Director, Central Sophisticated Instrumentation Facility, University of Calicut, to deliver the vote of thanks. Sir, please.
Yeah, just before that, I will honor, it's not only me, I am the convener, but we had 2 co-conveners also. And Dr. Professor Parthasarathi and Dr. Gomadi, but Dr. Gomadi had to leave yesterday because of some preoccupation, and now in this stage we will honor Professor Parthasarathi, who have planted the idea of this conference and workshop, and from where we have started with the Ponnada, and Professor Nasser will honor him. Sir, please come to the stage, please. Now I go into my duty. First of all, let me congratulate all winners of the various teams and the categories. We have, of course, have tried to plan it meticulously, because when we tried to even select the judges, we have made sure that none competed from that particular institution. So we selected those people who had no participants from that institution in that particular category, and that we have tried to arrange it so meticulously, and I am happy that all winners were, also we had some people who have presented well, but of course we were giving only 1 each in each category. It doesn't mean that other winners were not good, or sorry, other presenters were not good. It was equally good, but the best was given the prize. So best wishes to all those who have presented in that poster, as well as the other sessions, oral sessions. Now, just although the briefing has been done, again we had a total of abstracts 108 in this particular conference. We had invited lectures 5, lead lectures 7, short lectures 12, and the presentations were oral and poster 49 each, although there were some absentees. Now with each conferences we are trying to improve the way the conference is being run. I remember in 2014 or 13 or 14 we had the international conference on Syngiberraceae. Many of you may be knowing that our department, earlier Professor Sabu, who was in our department, former professor, was working with the Syngiberraceae, and we had an international conference in that area, and almost 40 to 50 international guests were here for that particular conference. And later we conducted the IAT conference, that is Indian Association for Angiosperm Taxonomy conference. And again in 2019 there was the IBS conference with the participation of around 150 people from outside Kerala. And again in 2022 we had the Indian Fern Society conference, again with a large number of participation. So with every conferences we are trying to improve. What our lacuna is there in the previous one, we try to amend that and come up with a better solution and try to improve upon. And whatever refinement what you see over here is the refinement what we have done through years. Now, first of all, let me thank the resource persons who have arrived from far and wide. You have seen already in the welcome speech of the first day, I already told people I have started traveling 2 days before the conferences, like Shabala, Professor Dangar, then even Sylvia,
And even one day before people have started, and all these people have traveled from far and wide, and I would like to, I am not naming individually because of short of time, because as you know, this academic event is not over at this point. Today afternoon we are moving for a field study, and that will be in Wayanad, and from there we will come back on Sunday, and Monday onwards we move into the workshop sessions. So 3 o'clock already we have scheduled for the field study trip, so that is why I am rushing through. So I am individually not naming any of the resource persons, but in one single sentence I would like to thank all the resource persons for taking the trouble to travel and reach from far and wide. And the conference participants, they are the foremost people, because the participation of the people from all area makes the conference the most successful. Because I have seen people conducting international conferences and national conferences with a representation from 2 or 3 states. Maybe if it is an international, it would be just one person coming from abroad. But this was truly, I feel that this was truly international and also national. Even we have representation from Shimla, so far and wide, Himachal. Imagine, from such far people have traveled. And in one sentence again, I am thanking the conference participants who have traveled so far and made this conference successful. Now next I would like to mention the team botany. I just put in these 2 words, team botany, because I cannot differentiate between the faculty, the office, the research scholars and the students, because we work as a team, and that is what you have seen over here. So I am not naming anyone individually, but just for names, I'll just mention about the committees over here. We had finance committee. We have already planned about this conference right from the month of August, and various committees have been working. The finance committee working under my convenership, food committee, Dr. Pramod, he's here he's here? No, again he has gone to take care of the food. Yeah, no, he's here in the back seat. And there is a committee working under him. Then for presentation there was Dr. Johnny Topil, our senior most professor is here, and there was a committee working under him, and press and publicity was by Dr. Reshmi is here. Yeah, just put up here. Yeah, Dr. Reshmi is there. And program committee, Dr. Manju C. Nair, she was always here in the forefront because she was conducting the program. And the registration committee under Dr. Shamina, and Dr. Shamina is here? No. Yeah, she's here. Dr. Shamina is here. And sponsorship committee. Sponsorship has come through the committee headed by Dr. Radhakrishnan here. And Dr. Pradeep was arranging the whole venue, the beautiful flowers and all these plants, whatever you see, everything arranged by Dr. Pradeep. He is here. Yeah. Then transportation committee was headed by Sunoj Kumar, Dr. Sunoj Kumar. He is here. So I am happy. All my faculty is here. Yeah. Then we had another committee. Yeah, that is the end of the committees. And I am not naming the whole committee because it's a long list. It constitutes of alumni from our own lab, and also constitute the other members or other students from entire department. Then I would like to make a special mention about Kalamandalam and our students for the terrific performance of their first day cultural program. It was really terrific, because you must have witnessed, I think some could not witness, Dr. Ashwini has missed that and some have missed that. But Kalamandalam, as I told, is specifically for the performing arts. They are students for the special university for performing arts. And when we invited them, they readily came over here, traveling 100 kilometers, and they performed before you. And after that, we had the performance by our own students. A special thanks to them. And I thank the press, especially our public relations wing. Shijit is here, our PRO. PRO is here. Okay, he was already, you must have made the note. He has made, you can see the excellent press reports which has come up. Already it has been put up in the board right outside the hall. So excellent press reports have come up. Sorry that everything has come up in Malayalam. Anyway, you can see the photos, etc. over there. Then the office support, I have to make a special mention. The office support, the section officer is here. Sridevi is here. Excellent support they are giving to me, because the whole finance side, the file side, you know how the process is being run in the university is very difficult. Every aspect, even for the flower arrangement, even for setting up the video wall, for everything the file has to move and take sanction right from the registrar or vice chancellor. And all this has been meticulously done. Okay. All this has been meticulously done by our office team. Sorry that I missed some committees in the first page. It is starting with abstract, so I turned to the next page. That is why I missed the major people here. Abstract committee was headed by Dr. Deepna, and I think she's in the audience. She's here. Yeah. Thank you for the excellent abstract book. You must have seen this excellent abstract which has just come up. And also the awards and certificate committee by Dr. Yusuf is here. Dr. Yusuf? Yeah, and was taking care of that. And accommodation committee, our great professor here, Santosh Nambi. The beautiful arrangements of the accommodation has been done. Cultural committee by Dr. Harilal and team. Dr. Harilal is here? Yeah, thank you, Dr. Harilal. So, again coming back, and the office support I was telling, the whole team, we have very limited office constituting of one section officer, one typist, and one assistant. And these 3 people plus one another assistant help also. These 4 people was taking care of the whole office functions, and it has been, everything has been done very meticulously thanks to the office. And the funding agencies, I have to make a special mention, because our Kerala Higher Education Council has given an enormous support to get the foreigners for this conference. And that is the Kerala State under the Erudite program. And also the DST has funded very good. The SERB has given us very good funds. And the KSST, again, the Kerala government's Kerala State Council for Science, Technology and Environment has given us good funds. INSA has given us fund. Also BRNS, Atomic Energy Department also has given funds. Also there was some sponsorship from private group, SRL, Chemind, Keagen, Amitech, BD Biosciences, Geol India, they also have given funds to us, special thanks to them. Food caterers already have been recognized, Fiesta, the printers from who has done the abstract book, Alois, and various supporting wings in the university. They were part of this, making this conference successful. The electrical people, instrumentation, the security and the vehicle department, engineering, garden and park staff, they all have worked tirelessly to make this a great success. And the team physiology, not the last or not the least and last, but they are the major people who have really done a great work. I would like to invite the team physiology in front of the stage, if they are here. I think again they are to the work back in the office. Team physiology, please come in front of the audience. Team physiology. Yeah, quickly call them, quickly, yeah. So, yeah, alumni also can join because they are all part of team physiology. Yeah. So these people have been a great support to me, and they were meticulously planning and handling the things very meticulously, and that is why everything has happened. And also the other students, but I have to make a special mention because they started the work 6 months back, and it was taken up by the students of Department of Botany later in the later course. So this constitutes the team physiology. This includes alumni, my own PhD students who have completed PhD under me, under my guidance, and also the present students are here. So special thanks to them. Yeah, a big clap to them. Yeah, you have to run, This short girl was assisting our promoter for the food arrangements. Okay, you can continue to stay there. Okay, and now our Pro-Vice Chancellor, he has agreed to be the chief guest of today's valedictory function. And yesterday when I was calling, he was returning from a long journey back. And around 12 o'clock only I got him, midnight, around 12 o'clock I got him over phone. And he said, certainly I am going to be there. Only you have to give me an excuse that I should be going to the church around 12:30, but I don't know. Still it is 1:15 and he's here. What happened? But maybe he has missed the time. Anyway, very happy that he's here on the stage till the end of this show. And I'm very grateful to you, sir, and also all others who are here on the stage and shared their memories. And once again, thanks to each one of you. Thank you. Thank you very much. And just I will make one or two announcements more, and with that our session will be concluding. One is that we will have photo of various committees being taken, but meanwhile others can start for the lunch, and we, the students and various committees, will take photos with the eminent plant physiologist over here, and some more to come up to the stage for the photo session. Then second is the certificate distribution will be done at the registration desk which you have already seen towards the left, and you can take immediately after the valedictory function. And there will be that traveler, 16-seater traveler going to the Calicut town at 3 p.m. and 6 p.m. So whoever wants to, either you are, if at all you are not going, if you want to visit the town, the vehicle will be going and you can move to the town and see the town and come back. But those who are moving for the field study should be here, because we will be starting at 2:30 from here. That is why we were rushing through these sessions. Okay. And the vehicles from University of Calicut campus to Luxora, that is the hotel where others were staying, will leave at 2 from here, and they should be very quick in their lunch, and 2 they should go over there, and 2:30 the vehicle will come back. So whoever are coming back in that vehicle should come back, because by 3 o'clock, between 2:30 and 3, we start for the field study. And of course, this was announced earlier. Till 28th evening onwards, the stay and food payment will be at the participants' cost. That is a sad part, but still I have to make the announcement. And if at all anyone wants the accommodation to be extended for 1 or 2 days, they can do so, but they have to give their names in the help desk. Only those people, we have to communicate with the guest house that they will be continuing, and accordingly they can be permitted, again on payment basis. Okay, thank you all. And now I invite the eminent plant physiologist to come up to the stage. We'll have some photo session, and just 10 minutes we'll be concluding that. Professor Shabala, Dr. Sylvia, please, and sir, Partha sir, please come to the stage. The certificates will be given after the lunch, so don't go for the certificates right now, because the entire team is here. You have the lunch and come back for the certificates. I think the first row can sit. You have to put more chairs. No, sir, sit please. Yeah, Dr. Babu, please come up to the stage. Dr. Vanaja is here. Dr. Vanaja. Dr. Dinagar. Yeah, please come to this. Sorry, there is for the official ending, we need the national anthem also. It was just missed. So we will play the national anthem, and after that we will go for the photo session. Yeah, please.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधितरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
After the photo of team physiology, we will have the photos of each individual committee. So committees, please wait, the conveners and the committee members, please wait. Joel, that is enough. That's enough. No. This right. No, I prefer to be here. No. You have to be there. The next is abstract committee is here. Abstract committee. Yeah, please. No, another three, 4 photos more, please. The certificate committee. Another 4, 5 snaps more. Yeah. Abstract committee. Yeah. Joel, one more chair. Yeah, certificate committee is here, awards and certificate committee under Dr. Yusuf, and others can join, the committee members. Yeah, only one is left. All others are in certificate distribution. Yeah, you can stay alone. That is it. Accommodation committee and cultural committee, I think they can come together, otherwise it will be too much of photos. Accommodation committee and cultural committee, please come together. Cultural committee, Dr. Hailal, please come together. Also, I think finance can join in this. Quickly. There's no time left. Yeah. First. Yeah, the food committee, presentation committee, press and publicity, program committee. These 4 committees, please come together. Yeah. Food committee, Dr. Pramod. ഇവിടെ വരുന്നുണ്ട്. ഈ 5 എണ്ണം ഒരുമിച്ച് വിളിക്കാം. അവിടെ കുറച്ച് തള്ളി നിൽക്കുക. നിങ്ങൾ 2 റോ ആയിട്ട് നിൽക്കുക. Yes. Please. Yeah. Now for the last one, registration committee, secretarial assistance, sponsorship committee, stage and venue committee, and transportation committee. Please, quickly come. This is the last photo. ഹൈറ്റുള്ളവർ സെക്കൻഡ് റോയിലേക്ക് പോകാം. ബാക്കിലേക്ക് കയറി. Okay, thank you. We have done with all photos. ഇവിടുത്തെ പരിപാടിയെല്ലാം കഴിഞ്ഞാൽ അല്ലേ? ഇല്ല, 10 ദിവസം ഇവിടെ ഉണ്ടോ? പ്രശ്നപ്പെടില്ല.